So before we start today, I would like to ask everyone physically joining us in this room to raise up for 1 minute of silence for the horrible event that happened yesterday in Azerbaijan. Unfortunately, there was an aircraft crash, and there were 14 soldiers lost their lives. So please raise up for 1 minute of silence to give respect to them. Thank you very much. You may be seated. Okay, thank you very much one more time for joining us today on the second day of the sessions. We are honored to have the first speaker, Gözde Demirer, joining us online from California. Gözde actually received her PhD recently. So she actually graduated from Koç University in Istanbul in 2015. Then she received her PhD from UC Berkeley. And soon she's going to join to Caltech, and she will have her own research laboratory. And today, Dr. Demirer is going to talk about the development of nano and biotechnologies for crop genome engineering. Dear Gözde, if you are ready, please share your screen with us. Thank you very much for joining us.
Hey, thank you. Let me get this set up. Sorry, not the screen. Are you seeing the correct view, or should I swap this place?
This is fine. You can proceed right now.
Okay. Okay, and can you hear me well as well?
We can hear you.
Okay. I think the connection is a bit slow, but
can you hear us? We can hear you. You can continue.
Okay. Okay. Yeah. Thank you, Emre, for the introduction, and I would like to thank all the organizers for inviting me to give this talk today at this workshop. I was going to start with a brief introduction of myself, but Emre has done that already for me, so I'll just skip to the talk outline. So today I'll be talking first about my PhD work, which was nanomaterial enabled biomolecule delivery for plant genetic engineering. And in the remaining time, I will talk about my current postdoc work, which is tomato genome editing for increased nitrogen use efficiency. So I would like to start by describing my PhD research. During my PhD, I worked on developing nanotechnologies to deliver genetic cargoes and perform plant gene editing for food security and sustainable agriculture. So I've done this research in the lab of Professor Marquita Landry at UC Berkeley, and I was involved with multiple projects during my PhD. But for this workshop, I'm going to focus on the two projects. First one is carbon nanoparticle development for gene delivery, and how we use this platform for the CRISPR-Cas9 gene editing in plants. So as you may all be aware and interested in this area as well, plant genetic engineering can address a lot of critical global problems that we are facing, such as in the field of food industry. We can create disease and climate resistant crops using plant genetic engineering. Similarly, engineered plants can provide advanced biofuels, as well as a scalable way of synthesizing therapeutics. And lastly, we can use the plants and engineer them for the remediation of our environment and sustainability efforts. I myself are most interested in the food and the environmental applications of plant genetic engineering. Even though all of these promise and excitement, there are still some challenges in plant genetic engineering. And one of the challenges that I've focused is the biomolecule delivery into plants. And the tools for biomolecule delivery and transformation in plants are very limited. And that's mostly because of the cell wall that's around these plant cells that acts as a physical barrier for the cargo delivery into the cells. So here I'm showing the size exclusion limits of certain barriers around the plant cells. So if we look at the lipid cell membrane, these membranes are able to transport across anything that is smaller than 1,000 to 800 nanometer. But when we look at the cell wall, we see that it can only pass molecules that are smaller than 20 nanometers passively. So which was a main barrier of delivering particles inside the plant cells. So given this limitation, the most commonly used method in plant gene delivery is the bacteria, agrobacterium, which many of us are using to do transformations in plants. This method is able to deliver DNA, but to a limited plant host species. So our crop of interest may not be transformable with agrobacteria. And it can only deliver DNA cargoes, so we are unable to deliver molecules like RNA and protein. And that DNA cargo that we deliver gets inserted somewhere randomly in the plant genome that causes a lot of regulatory restrictions. So the current GMO crops we have in the market today, in average, took 13 years of research, and it had a very high cost of 136 millions. And around 40 millions of this cost was due to the regulatory pipeline. So when I started my PhD, motivated by the impact of plant gene editing and the lack of availability of delivery tools, I wanted to develop a plant delivery technology that can work in a plant species independent way and efficiently so that we could use it in many different crops, as well as we wanted to design it so that it can have less regulatory burden by making it abiotic and non-integrating. So to achieve this, I used nanoparticles, and nanoparticles have been used in many animal and mammalian systems for gene delivery. And recently, as we all know, we have used it for COVID vaccine delivery using the lipid nanoparticles. So in this project, I used a material called single-walled carbon nanotubes. So this is a material based solely on carbon. And in the center, we have this one called SWINT. So that's single-walled carbon nanotubes. And the reason why we wanted to use this is that if you look at its diameter, it has 1 nanometer in diameter. So it's very small in this dimension that could enter through the cell walls, but it's also very long. So this is a sphere. It's a cylinder that's hollow in the inside, but very long. So this means that it has a very large surface area that can load a lot of molecules like plasmids and proteins on the surface. When we compare this single-walled carbon nanotube, for example, to other carbon materials like this buckyball, which is just a sphere of carbon, it is 1 nanometer in every dimension. So this one is also very small, but it has a small surface area, so we can't deliver enough proteins or DNA using this type of a material. And when we look at molecules like graphene, that has a very high surface area, but it's also very large in each dimension, so it will be hard to enter the plant cells. Given these abilities, as well as its high stiffness, we decided to use single-walled carbon nanotubes to deliver genes into plant cells. So how this works is that for the initial development of this system, we wanted to load these carbon nanotubes with a plasmid that is going to express a GFP gene so we can easily detect it. So we used a very simple plasmid that's driving a GFP expression with a common 35S promoter. And we introduce these into the leaves of plants. So here we are looking at a Nicotiana ventimiana tobacco plant leaf, and it's injected with a needleless syringe, just like we do agroinfiltrations. And once it's inside the plant tissues, we would expect it to be able to transport across the cell wall and the plasma membrane. And once it reaches the nucleus, this plasmid DNA will just make mRNA and the protein of interest, so in this case, GFP. So before looking at the protein expression, we wanted to see if these DNA-loaded carbon nanotubes can enter the plant cells. And for that, we started looking at the protoplasts. So protoplasts are leaf cells that we can extract from the leaves and remove their cell wall using enzymes. So this is an initial system that we wanted to test. Given it doesn't have a cell wall, it's an easier place to begin with because the nanoparticles would only need to pass through the cell membrane in this case. So here we are looking at an extracted protoplast here on the left, and we stained its nucleus using the stain DAPI that's shown in blue, and then we have the DNA labeled with Cy3, which is a red fluorophore. So in this case, we are delivering the free DNA without carbon nanotubes, and in this case, we don't see any internalization of this DNA into the protoplast. When we deliver the same DNA with carbon nanotubes, then we see that the DNA localizes both in the nucleus and the cytosol of this cell. So this shows us that the DNA carbon nanotubes can enter the plant cells when they don't have the cell walls. So next, we looked at if they could also enter into the insect leaf cells. And in this case, I'm showing here an image from a tobacco plant, and this is just a reminder that when we look at the epidermal cells of these plants, there is a huge vacuole in the center of this cell. So then the cytosol is pushed to the cell wall. So here the green contour is where the cytosol is, and this circle here is the nucleus. So keeping this in mind, when we look at the DNA carbon nanotubes that are tagged again with the Cy3, we see them inside the cells, both in the cytosol and in the nucleus. So this shows us that the nanoparticles are able to carry this DNA into the nucleus, where it will then turn into mRNA. So after seeing that these can enter plant cells, we looked if they could actually express their cargo, which was GFP. So in this case, we're looking at expression in the arugula protoplasts. So on the top, again, there's the free DNA without nanoparticles. It's not able to enter the cells by itself. But when we express it with carbon nanotubes using the 35S GFP construct, we see it in the cytosol and in the nucleus. And we have also expressed a GFP that was targeted to nucleus. So in this case, we see GFP only in the nucleus of these cells. So after seeing expression in the protoplast, we then looked at expression in the leaves. So here we are looking at leaves after 3 days of infiltration with our particles. And this first one is the tobacco plant again, Nicotiana ventimiana. We have also done this in arugula, wheat, and cotton. And in all of these cases, we were able to see GFP expression in these plant species. So what we saw is that this GFP expression is transient. So the high levels of GFP we observe using confocal at day 3 completely disappears at day 10. And this is at the protein level. We could also look at the mRNA levels of this GFP protein, and we see that it's been stopped making at day 10. So by day 10, there are no more GFP mRNA. So from this, we hypothesized that the proteins could be expressing transiently from the vector, and the vector does not get integrated into the plant genome. So just to show this with a schematic, here when we deliver the plasmid with carbon nanotubes, it enters the plant cell and it makes the protein from the plasmid itself, but this plasmid does not get inserted into the plant genomic DNA. And over time, in 10 days, when the cells divide and this plasmid DNA degrades, we don't have any more plasmid DNA in those cells. In this case, we don't get any more of the protein expression. So this is different in agrobacteria. When we deliver plasmid or tDNA in this case into the plant cell using agrobacteria, the protein is made from the tDNA itself, but then also that tDNA gets inserted into the plant genomic DNA. So even after the plasmid DNA is degraded and diluted in the cells, we still have GFP expression from the genomic DNA insert. We showed this also with the qPCR in agrobacteria, that the mRNA levels will keep stable over a long duration of time as it's expressed from the plant genomic DNA. So to validate this hypothesis even further, we used a method called droplet digital PCR. So this is a very sensitive method that can detect even one integration event out of 500,000 integration events. So when we treat our plants with the carbon nanotubes and agrobacteria, and after a certain time, when we look at the genomic DNA, we could see if there are any integration. So in these plots, for example, in the agrobacteria, we are looking at all the genomic DNA we collected from one leaf, and each green dot is an integration event. So in a full leaf, we have a lot of integration events in agrobacteria delivered sample. But when we deliver the same DNA with carbon nanotubes, there isn't any integration event. So this validated that the DNA, when we deliver it with nanoparticles, is not available for integration, and it only expresses transiently, which lasts for 7 to 10 days. We wanted to make sure that this tool will be applicable in many different species. So after our initial experiments, we have also tested carbon nanotubes in various different dicots and monocots. Here I'm showing some results in arugula, Arabidopsis, wheat, and some hard to transform species like the dicot cotton and the monocot sorghum, which was very exciting to see. All of these are leaf samples. We have also shown that we can get expression in callus, stem, embryo, and pollen samples. So it also works with different types of tissues, which will be important to do heritable edits going forward. Also, I want to mention that at the concentrations we use them, they are not toxic to the plants. So here we are looking at a leaf that was infiltrated with water, 2 different amounts of carbon nanotubes, and as a positive stress control, we infiltrated SDS. So we could see that the SDS area is having tissue death and necrosis, but the carbon nanotube areas are visibly fine. We could also look at the rates of photosynthesis in these areas. So this is a way of quantifying photosynthetic efficiency. So in the area where we infiltrated the SDS stress inducer, we see low values of photosynthesis efficiency. That means that that area is affected. But in the area where we infiltrated the carbon nanotubes, the plant is able to do photosynthesis just like a non-treated area. In addition to these, we have also looked at some molecular markers of stress in plants. So we picked this gene that's a marker for stress in plants. It's reactive burst oxidase homolog B gene, which is upregulated when plants are under stress conditions. So we could see that in the SDS infiltration, the plants are highly stressed as expected, but in our carbon nanotube treatments, the stress levels are very low, and it's very comparable to just the buffer infiltration. And we have also done some RNA sequencing results that I won't be able to share today given the time restrictions, but we were able to determine this PR1, pathogen response 1 gene, as a marker to look at the carbon nanotube toxicity. And we could see that this toxicity depends on the concentration of the DNA carbon nanotubes we use. At low concentration, for typical experiments, we would use 1 milligrams per liter. The toxicity is low, comparable again with water infiltration. But as we go higher concentrations, its toxicity is also increasing. So that's something to keep in mind when we are using higher amounts. So to summarize this part so far, we were able to develop a delivery tool that is so far species independent, efficient. It is completely abiotic. It doesn't use any virus or bacteria parts, and it does not integrate DNA into the plant genome. We have shown that it has a good efficiency. So it's more efficient compared to a gene gun where we get a larger cell population expressing. But it's less efficient compared to the agrobacteria. So we usually get maybe 30% of what we get when we compare it to agrobacteria expression. So it's somewhere in between the efficiency of a gene gun and an agrobacteria. And as I mentioned, it doesn't cause integration. So this was the first time delivering nanoparticles into plants that didn't use an external force like a gene gun method, and it addressed some of the bottlenecks of DNA delivery to plants. So next, we wanted to use this developed tool for CRISPR gene editing. So all we needed to do is just to replace our GFP plasmid with a plasmid carrying CRISPR parts. So in this case, that would be a Cas9 protein and a guide RNA coded in this vector that we can load onto the carbon nanotubes. And for initial experiments, we designed our guide RNA for gene knockout to cause small insertion and deletion events in DELs, and to see if we can use carbon nanotubes for gene editing as well. So we have done all of the gene editing studies in Nicotiana benthamiana in the tobacco plant. So I'm going to show a couple of experiments. The first one we did, we used a Cas9 expressing transgenic tobacco. So then we would only need to deliver guide RNA plasmids. So it's an easier place to start again. And we have also done the similar experiments in the wild type tobacco and in the GFP expressing tobacco so that we can knock out the GFP expression. And in these cases, we would need to deliver plasmids coding both the Cas9 and the guide RNA. And in these initial experiments, we targeted the phytoin desaturase PDS gene. So this is a visible gene when it's knocked out that the plants turn white. So it's an easy way to phenotype the edited plants as well. And initially, we used 3 methods to quantify gene editing. So the first one is Sanger sequencing and indel analysis using TIDE. This is, I think, the method that is the, first of all, easiest, less labor, and it's more quantitative and precise compared to other enzymatic-based methods. So the other method that we used is this T7 endonuclease 1 enzyme that recognizes mismatches in the genomic DNA caused by the CRISPR editing, and it will cut the DNA in there. So in the edited samples, you will end up with multiple bands when you run this on the gel. In my experience, it gives a higher background. It's a bit more labor intensive, and you need to be careful designing your guide RNA primers because you don't want to center the guide RNA. The other method that's also enzymatic is the restriction site mutation assay. So in this case, when you're designing your guide RNA, if you design the guide RNA to mutate a restriction enzyme site, so in your mutants, the restriction enzyme site will not be there. So when you extract the genomic DNA and add the restriction enzyme in the wild type, it should be cut into 2 pieces. But in the edited one, because you don't have the restriction enzyme site anymore, you will get a 1 band. So then it's again an enzyme running on the gel type of assay. It restricts highly the types of guide RNAs you can design because it also needs to target the restriction enzyme site in addition to your gene of interest. And it's again, because it's enzymatic, it's a bit more labor intensive. So we used all these 3 methods to quantify our editing efficiencies. So here I'm first showing the editing results of the Cas9 tobacco. So that's the transgenic tobacco that we only needed to express a guide RNA vector. So this guide RNA was designed in this paper earlier, where they also knocked out the PDS gene. What we did, we loaded this plasmid onto the carbon nanotubes, and then we did a heat treatment. So the plants were in 22 degrees for 24 hours, and then they were at 37 degrees for 24 hours, and we rotated this cycle 5 times. And we were just trying to increase our editing efficiency, as Cas9 has higher activity at higher temperatures. So I'm first going to just show the results for each of these editing, and then I'll go into the details of quantification a little bit more. So in this case, our editing percentage when we use carbon nanotubes is around 15%, and this is indel generation efficiency. And in the case of agrobacteria, it's around 40%. So we could see that carbon nanotubes are able to cause gene edits. They are less efficient compared to agrobacteria in this case. And when we do the similar assay in the wild type tobacco plants, in this case, to knock out the PDS, we expressed a 35S driven Cas9 and a U6 driven guide RNA. It's the same guide RNA from the first assay. And then we did the same heat treatment. This time we see more or less comparable editing efficiencies between the carbon nanotubes and agrobacteria. So carbon nanotubes are causing 2% editing efficiency, whereas we have around 3%, 2.8% for agro. So these efficiencies may seem low, but this is just the editing in the leaf. So we are not selecting any edited cells. So when we collect genomic DNA, we get low of wild type that is not transformed, but 2% with carbon nanotubes we think it's a really nice achievement, because just to remind you that in this case with carbon nanotubes, it's a completely transient expression of the Cas9 and guide RNA. So this vector did not get integrated into the plant genome. The Cas9 and guide RNA was only there for 7 to 10 days. They did editing and then they disappeared. Versus in the agrobacteria, these cells will have the Cas9 integrated. So in the next generation, say this is a heritable transformation, we would likely need to remove the Cas9 and do segregation. So it has advantages of being able to do same or similar editing efficiency without integrating the Cas9 vector. And in the last case, I'm going to show the editing of the GFP gene. So here we had the GFP transgenic plant, and we wanted to knock out the GFP in that plant. And this time we designed 3 guide RNAs targeting different parts of the GFP gene, just to study if the location in the gene makes a difference. And again, the same heat treatment. So we see varying efficiencies with different guide RNAs from 4 to 8%, but it wasn't a very drastic difference between different guide RNAs. All guide RNAs were designed ideally in terms of their sequence. And what we also did here is that we knew that the carbon nanotubes are not able to travel too far from where we infiltrate them. So they stay typically local around the infiltration area. But when we collect genomic DNA from the entire leaf, that just dilutes the amount of edits we get. So this time we decided to just look at the entire leaf area as well as just locally. And what we see that if we look around the infiltration area, we have 5 times higher editing efficiencies. So again, this is something to keep in mind when we're using these nanoparticles. The editing events will be around where we infiltrate them, and it's not going to go further away from that location. So these are the broad results that we obtained using carbon nanotubes for editing. But I also wanted to show how we actually did the verification and quantification of this using those 3 assays that I mentioned. So I'll just show this for the transgenic Cas9 tobacco plants. So in this case, the first I'm showing is the restriction site mutation assay. And then initially we were doing these without heat treatment. So we are looking at the without heat treatment result here. So this is the amplified PDS gene before we add the restriction enzyme. So it's not cut here. We have the PDS gene sequence. So we have a non-infiltrated sample, 2 agrobacteria infiltrations, and some negative Cas9 plants that is not infiltrated or infiltrated as egg rows. These are negative controls because when we don't have Cas9, we should not get any editing in these plants. So then what we do, we treat this PDS gene with the restriction enzyme. So in the case of non-infiltrated samples, so there is no editing in this one, you could see that the band at 450 disappears completely and you have 2 cut bands. This means that the sample is non-edited as there aren't any gene copies that are resistant to the restriction. Whereas in the edited samples, we could see that because the editing is not 100%, we still have cut bands, but we also have a restriction enzyme resistant band at the top. This means that this 15% of the cells did have the edit. So this is how we quantified, and in the negative controls where we didn't have Cas9, there was no editing. So this is without the heat treatment. We thought that these 10 and 15% were a little bit low. So then we did the heat treatment and did the similar assay. So what we see here in the case of agrobacteria, the editing efficiency was 44%. So almost like half of it was not cut and then half of it was cut. So we could see that the heat treatment really increases the efficiency. And in this case, we included our carbon nanotube samples as well. So we saw that there is a faint band at the top. This corresponds to 13 to 14% editing in the carbon nanotube samples. So this is how we quantified using the restriction site mutation assay. Similarly, we could use the T7E1 assay. So this is the one where we can use this enzyme to cut the edited copies of the genome. So in the case of non-infiltration, we don't have any cut bands. So this means it's not edited. In the case of agro-positive and the carbon nanotube positive control, we have cut bands. But as I was mentioning, there is also a lot of background bands, and it's not always the cleanest way of identifying gene edits. But it gives similar editing efficiency with the restriction site mutation assay when we compare them. And then the last assay that I think is the most quantitative and always gives cleaner results is the Sanger sequencing of these PDS amplicons and running a tight analysis on them. So here we are looking at one of the agrobacteria samples, and it shows us that like 38% of the genome is not edited, but we have total of 35% that is edited, and it also shows the breakdown of these edits. So it tells us that like there is 16% plus 1 insertion events, 7% minus 1 deletion events. It gives us a breakdown of all the different types of edits. And we can similarly look at this for the nanoparticles, where we see a total of 15% editing efficiency. Most of it, 10% of it is a +1 event. So these are the 3 methods that we used to quantify our gene editing when we delivered CRISPR cargos with nanoparticles. So this was a more or less recent work, and there is a lot more ongoing work that's happening in the Landry lab right now. So just to summarize, we got the highest editing efficiency in the Cas9 transgenic plant. We had lower editing efficiency in the wild type plants, but comparable between agrobacteria and carbon nanotubes. Guide RNA design is important, but with today's softwares, it's easy to get optimal guide RNAs designed. And closer to the infiltration area, we have higher editing efficiencies. So what's happening right now is optimization of the carbon nanotube amount and infiltration conditions. We have been trying the Gemini virus replicon vectors to increase editing efficiency. We are improving our constructs by optimizing for codons and promoters. And lastly, we want to look at the off-target effects using a whole genome sequencing. And we are trying to translate all of these editing we have done on leaves into heritable transformations using developmental regulators. So as I mentioned, we have interest in being able to do these edits heritably. Maybe flowers and pollens and shoot apical meristems could be nice tissues for this. In addition, I think it makes sense to use carbon nanotubes for transient expression of morphogenic regulators and developmental regulators. So these, when they are expressed transiently, together with the CRISPR cargos, will give rise to gene edited new embryos. So it could be a fast way and maybe a tissue culture-free way of generating edited plants using the nanoparticles for transient expression of these tools. And more on the chemistry side of the things, so far we have only developed methods for the plasmid delivery, but we also have interest in delivering Cas9 as a protein and a ribonucleoprotein complex as well. So we are trying to develop some chemistries for these different cargoes. So all of this work was done in the Landry lab at UC Berkeley. I have a lot of people to thank there and a lot of funding sources for this work. And I did have a Schlumberger Foundation for my PhD work as well. So I would like to acknowledge all of these people and funding sources. So in the remaining 15 minutes I have, I'm going to talk about my postdoc work that I'm currently completing. I'm doing this in Professor Shibam Brady's lab at UC Davis in Plant Biology Department, and we are hoping to engineer nutrient use efficiency of tomato plants, again, through CRISPR gene and epigenome editing. I'll start by saying why I'm interested in studying plant roots and nutrients. And that's because we and the plants are facing changing environmental and climate conditions with increasing temperatures, excessive availability of water or drought, and a lot of new emerging pests and diseases. All of these decrease the crop productivity and yield. Given these changes in the environment, as well as the increasing human population, we need to increase the crop productivity, but we need to be able to do it in a sustainable way. So over the last century or so, we've been using a lot of chemical fertilizers in an unsustainable way, just to increase the crop yields. But these fertilizers had negative consequences on the environment. So if we could understand how plant roots respond to the environmental conditions, the availability of nutrients, so that we could engineer these root systems more efficiently to be better adapted to the changing conditions, as well as to support the growth of more productive plants using less nutrient fertilizers, that would be a very impactful way of going forward with some of our gene editing studies. So in my postdoc lab, I focused on nutrients, and I specifically worked with nitrates and phosphates. These are essential nutrients for plant growth, but their excess usage has been causing a lot of ecological and environmental consequences. So we need to understand how plants use these nutrients so that we can do precise edits to use as minimal fertilizers as possible, but still have higher crop yields. So this is what I've focused during my PhD. I'm going to try to not go into so much detail here, but I'm initially focusing on the nitrogen metabolism regulation. And I know this looks very complex, but what I'm showing is actually, this is in Arabidopsis. This is a gene regulatory network in the center showing transcription factors regulating a lot of different genes. And this is only for the nitrogen. So all I'm trying to show here is that this is a very complex network of genes and proteins in Arabidopsis. And as we all know, Arabidopsis is one of the smallest genome plants with only 135 million base pairs. And when we translate to tomato and many other crops, we have huge genomes. For example, in the case of tomato, we have 950 million base pairs. So you could only imagine that how this nitrogen network is going to look in tomato and how hard it is to understand these interactions, yet alone edit them. So this project focuses on trying to understand some of the key transcription factors in the nitrogen network. And it's a collaborative project between Brady Lab and Nicola Patron's lab at UK, where we are looking at these transcription factors and how they regulate nitrogen metabolism, both in Arabidopsis as well as in tomato. So our aims is to look how much of this pathway is conserved between these two species, because most of the research is done in Arabidopsis that aims to increase the nutrient use efficiency, but we need to understand what we can translate in between Arabidopsis and different crops. So how we do this, so our aim was to create the single and higher order mutants of all of these transcription factors in tomato. And because transformations take a long time in tomato, we use a different system. that is based on hairy roots. So if you're not familiar in hairy roots, that's a fast way of doing CRISPR transgenics or other transgenics in crops like tomato. So we start with cotyledon, a piece of leaf, and we transform it with a bacteria called rhizogenes, Rhizobium rhizogenes. So this bacteria carries a Cas9 and guide RNA, as well as a kanamycin resistance. And after a couple of weeks, we started to get these roots called hairy roots from the transformed leaf pieces. So each of these roots will have a different genotype. These are all independent transformation events that we select on a kanamycin plate, and then we can propagate them on plates like this. So this is how it looks. You start with leaves, and then they start giving these root pieces. You can transfer an individual root onto a plate and culture it like this. So this is a mutant of like one of the transcription factors, for example. And we can genotype them, and we see a lot of different types of CRISPR edits, large deletions in some cases, or indels. In tomato, we typically use 2 to 3 guide RNA per gene to make sure we get nice edits. So in this case, we did all the single and combinations of mutants in this network. So you can see we have 7 transcription factors. So we had constructs that contained 21 guide RNAs in some cases. And how we do such complex editing and cloning is using this multiplex mutagenesis strategy that was developed by Pollack et al. So this is called loop assembly. So it's a way of cloning very large vectors with a lot of different parts. If you look at it, it has the region where you have the guide RNAs, Cas9, resistance, and some selection as well. So technically, it gives you a way of doing a limitless amount of insertions. So as I was saying, we have all 7 transcription factors mutated. So that means 21 guide RNAs in this one vector that we were able to clone in a very easy and fast way. It has a marker, kanamycin, for the transgenic plant and root selection, and it also has a seed marker. So this is fast red. It turns the edited seeds into red, so we can easily pick the ones that are edited with a dissecting microscope. So what we are doing currently with all these lines, because we have so many lines, we are genotyping them by deep sequencing of PCR amplicons using a facility at Harvard Medical School. So they are giving us the types of edit and the percentage of edits in all cells we have. All lines we have. We are phenotyping these mutants under different nitrogen conditions, and we have already identified some promising lines where the knockouts gave us a higher nitrogen use efficiency and nitrate responsiveness. So we were able to see some of those transcription factors and interactions being able to grow these tomato with less nitrogen input. So we are, because these hairy roots are not real stable transgenics, we are in the process of creating stable mutants of these promising lines through callus-induced mutagenesis, traditional way of doing the transformations in tomato. Okay, so with that, I would like to also thank my postdoc lab, Shubham Brady, and all of our collaborators at Berkeley, Israel, and UK, especially Nicola Patron's lab. And thank you all for listening. I'm happy to take questions now.
Okay, Gözde, thank you very much for this amazing talk. So right now we can take a few questions from the physical participants first, then we will go to the online ones. Yes, please. Can you come to the stage to ask the questions? Thank you.
Hi, Gözde. I am Nihal. It was a very nice talk. I'm Nihal from Sabancı University Nanotechnology Research and Application Center, and we want to replicate your research about carbon nanotube and the delivery of the vector to several actually crops. My question is a bit technical. I want to ask you what other ligands that you can use instead of PEI to attach plasmid onto carbon nanotubes? And I have another question after that.
Okay. Yeah, great question. Thank you. Yeah, I didn't go into the details of the chemistry today, but PEI was the positively charged polymer we used to be able to condense the DNA on the carbon nanotubes. And we are trying to switch it with different types of positively charged molecules because the toxicity I showed in the RNA sequencing data, that is caused by the PEI. So when we use high concentrations of PEI, we end up getting a lot of toxicity. It's more manageable in the leaves, but it became a problem when we were working with the protoplasts and tissues that we tried to regenerate. So we are trying a huge library of positively charged polymers. I think one of the ones that worked as good as PEI is polylysine. So that's an alternative that I would suggest. It's a bit more expensive than PEI, but it has a lower toxicity, significantly lower toxicity. So that's an option. I'm not sure of any other one that has shown success yet, but I could check it out and let you know. So this work has carried on after I graduated. So I know that they've been looking at different molecules as well.
Okay, thank you. And I have
sorry, there's another one actually. There's
sorry.
chitosan, I think, as well, that they had, yeah, they had success loading DNA.
Okay. And also, can you tell me the maximum size of the plasmid that you can use for the delivery?
Yeah, that's also a great question. So it's more efficient with smaller plasmid. So the initial optimization we have done, they're all with 4 to 5 kb plasmids, which is not realistic really with when you're looking at CRISPR plasmids with selection markers. We did get expression from things up to 12 to 14 kb with a lower deficiency. I have not tried anything larger than that.
Okay. Thank you so much.
We can take one more question over here. Sadia, please.
Hi guys, a great talk. Thanks for your talk. And my question about, you told you transfer them like infiltration, but have you tried, and you tried in with leaves, but have you ever think about the regenerate plants from the leaves, like the?
Yeah, we haven't tried that. Yeah.
That a morphological gene technology that we can use them for the increased regeneration and for the somatic embryogenesis, and we can regenerate plants from the leaves.
Yeah, exactly.
So
that's
the kind of study.
Yeah, I think that'll be a great way to go forward. We haven't tried that. I think it'll be easier than like trying to regenerate from callus. I think because we know we can do the leaves, so then we could try to regenerate from leaves. What I've been suggesting to do for future is to transform the shoot apical meristems, especially in tomato, they are pretty visible. So you can inject the carbon nanotubes together with the developmental regulators into the shoot apical meristems in the leaf axillary meristems, and then hopefully you'll get new branches that are edited that you can collect seeds in the future. So that's what I've been trying to develop in tomato right now with the shoot apical meristems, but I haven't tried the leaf regeneration.
Okay, because you thought you can transfer them to infiltrate with leaves mostly. I see your presentation, but you can use ITEX in immature embryos as well, right?
We haven't tried embryos yet. So I think you may need to optimize for embryos, like the concentrations and the protocol, but you could just, you can put it in a syringe and inject into the stems or the meristems, for example.
You can regenerate transgenic plants from the leaf explants, instead leaf explants. It would be interesting to try in meat like this, I thought.
Yeah, I think so too. I think it'll be really exciting to try.
Okay, thank you.
Thank you.
Now we will take one more question.
So, one part of my question is already asked by Haita, so I'll ask the second part. So there are reports that if you dip roots in carbon nanotube, probably they can take up the DNA and that goes into the upper parts and look maybe very low efficiency, but I mean, if it works, then it works. Your comment, please.
We have not done it. We have not tried that either. I did see those papers, yeah, through the roots, and I will. and then it increases the germination rates. So I think there are some benefits of having carbon nanotubes in the roots and in the germination media, but we haven't tried delivery through the roots. So yeah, I don't have much to say on that because we haven't tried it.
Okay, we have one more question from the physical audience.
Hi, Josda. Thank you very much for your very interesting presentation. Actually, I am not expert on the nanotechnology, but I know that in the medical system, there is nanoparticle-based tissue-specific delivery system. Is there any tissue or cell type specific gene delivery system in your lab or in other part?
Yeah, I think that'll be really nice to develop. I think that's one of the advantages of the nanoparticles that we can take advantage and use in plants as well. So I think the easiest would be to target by pH, because we can have nanoparticles that are responsive to pH. So then like the cell wall pH is lower, vacuole pH is lower. We could potentially target those compartments. And we can also take advantage of targeting peptides on the nanoparticles, because we know sequences of targeting peptides for the chloroplast and mitochondria. So we could attach those peptides on the carbon nanotube surface to put them into the organelles, but that's something we haven't tried yet. I think we did try to attach some peptides on them, but the stability was an issue. So the protocol, we need to optimize it a little bit more. When we have like both the DNA and the targeting peptides, the complex was not stable enough, but I think there's a potential in investigating those.
Thank you very much.
Okay, now I will also look at the chat box to see if there are any questions from the online participants. Let me see that. Okay, it seems that there are no other questions there. Dear Gözde, thank you very much for joining us. I know it is too late already there. Have a good night, and we appreciate your presence here.
Yeah, thank you so much, and thank you all for great questions. Have a good rest of your day.
Thank you, bye. Okay, dear participants, we will continue with our next speaker, dear Dr. Sadiye Hayta. She's together with Mark. They are from John Innes Center, and Dr. Sadiye has been working on wheat transformation, and she has developed an interesting, very important and very efficient protocol for the transformation of the wheat. And I think today she will talk about this transformation efficient protocol as a case study. Thank you very much for joining us. Just bunu şöyle yapacağız. Şunu bir açayım ilk önce. Ondan sonra da buradan ne oldu? Buraya nasıl geri geçeceğiz ki acaba? Bunu tekrar nasıl açacağız Zoom'u? Orada olduğumuz için. Pardon. Evet, bir de şunla mı açılıyor? Lazer. Buyurun.
Thank you, Emre, for introduction, and good morning, everybody. I'm Shadi Haita and I work at Genomics Center. And today I'm going to talk about our transformation system. I work at Genomics Center, and Genomics Center is a part of Norwich Research Park. And I'm also working in the crop transformation group and responsible for wheat transformation and genome editing. In wheat, we've done so far gene overexpression studies, gene complementation, RNA interference, and mostly genome editing. Actually, yesterday, Mark explained wheat is one of the most difficult crop and it has got very big genome. And with traditional breeding and mutation breeding, it's very difficult to use in wheat because wheat has got big three subgenome. If you just mutate it, if you cast up one mutation in one subgenome, can you hear me or shall I just stop? Okay. Can I use this microphone? Okay. Traditional breeding in wheat very efficiently. Second method, you know, as everybody knows, it's mutation breeding. But again, you need to knock out three because mutations, because which really we don't want. Polyploid plants like wheat is the crisp before. And if it is or maize, if you knock out one genome, and you can see that this kind of 21% increase in grain weight. But in it, you need the knockout three. You can see 6% if you do double mutation, 11%. And if you knock out the three subgenome, you can see the 20% grain weight. Is problem with okay. Our strategy in wheat is depend of our target because we are working in wheat researchers at Genomics Center, also not only Genomics Center, other wheat researchers as well. Usually they send our target to us and we just check them. We can depend on their target, base editing, multiplex editing, even the prime editing, but mostly of course knockout. And we first, yesterday Mark explained all of them, but first we design our guides. And usually we target the first intron, and mostly we use 2 guide RNAs.
I think the Zoom people cannot see us.
Again, I was talking about our strategy in genome editing in wheat. And usually when we design guides, we use the 2 guide system, but it depends on, again, our targets. And we can use now 6 guides in the wheat genome editing. And for each target, we use 2 construct, and each construct got the 2 guides usually. And then after we prepare our construct, we transform into wheat. In wheat genome editing, actually also all the genome editing is one of the important factor is the good sequence data and tools for designing guides. If you haven't got any good detailed sequence data, you can't design it. And in wheat, again, big genome and the good sequence data really revealed is recently, 2018. I will tell you a bit later more details. And second important factor is an efficient delivery system. It can be biolistic or agrobacterium, we use agrobacterium transformation protocol. And also screening is also the important factor, especially like the wheat, polyploid wheat plant, it's more challenging and you need to produce a lot of plants to screen it. In wheat, the good high quality genome is just released 2018 and it was in Chinese Spring. And years we use the Chinese Spring as a genome sequence for design guides. And we transform filter. But recently, especially in the last 8 years, we a lot of high quality hexaploid or tetraploid with genome release, and we can now use it efficiently in our CRISPR world. Can I use this one now? Okay. As I previously say, I work with researchers and they send us their genes, and we work with them to assess their gene function. And after that, we see if we are working in Kronos or Paragon with each variety they send, we design the guides for them and transfer them to in vitro. Second factor is efficient delivery system, which considered one of the most difficult crop to transform, and it is very genotype dependent. The first transgenic plant is obtained in early 90s, while Agrobacterium-mediated transformation remained around 5% after that. And there is a remarkable protocol in 2015. It is a patented licensed protocol, and it requires special training and vector, so it can be used very efficiently. Our open access protocol is published 2019. It is an efficient protocol, but still it is genotype dependent. And 2020, with Juan Dabrowski-Raab in UC Davis, we published a protocol. It's increased the efficiencies in with transformation. Also, it's increased regeneration, transformation efficiencies, also reduce the genetic dependence drastically. In our transformation system, we use agrobacterium transformation system, and we use spring wheat variety filter. And our efficiencies is good for the filter's efficiency. It was 33%, and the Kronos we can get 10%, in Cadenza 5%. It's great for the wheat varieties. But our transformation system, like not ours, the others as well, affected from a lot of factors, and wheat is more sensitive than other crops. Even the growing conditions, small change, or growth donor plants had a big change, these factors are dropped drastically. We use Golden Gate vector system for our transformation protocol. It allows us to clone quickly and efficiency. Also with the combination of PGG vector with our helper plasmid, which contains a VRG, give us higher transformation efficiencies. We use gas reporter construct for initial our experiments for optimization of transformation protocol. And if you got interested in our transformation system and constructs, you can contact the Mark, or you can find our constructs all in HGene. In our transformation protocol, we use immature embryos, and this is the stage the transformation affect a lot. If our plant material is not healthy or in wrong stage, or transformation efficiency is affected drastically. We have a centrifugation stage before co-cultivation. Leaf samples for DNA extraction, and we check the edges. And I just want to give us some of examples from our transformation studies. In this study, we Thank you. A second example I would like to share with you today is our collaboration with Brand Wolf. He's now in cast and he's actually, if he interested, he's looking for postdocs and PhDs. And this is, it is in stem rust resistance, and we just check the resistance transgenic plants and compared with wild type, you can see that they are sensitive and they are more resistant. And it's patented now. Now we start to use a protein fusion growth regulating factor, and is it the cofactor GRFT in our transformation efficiency, and now we can, and also it shortened our transformers is not affected from other external factors. My first results from the paper we published drastically increase and they don't affect any other external factors. Now our transformations, running our transformations efficiency around 77% in filter and 70% in Kronos, especially Kronos gain more benefit from this system. We can transform mature embryos. I just tried, so we don't need to grow plant matters in in CRs or glass house. So we just pick up from shelf and we can transform them. And efficiency, it was my first try, and efficiencies were in 9%. Also now we know that we can transform cyclic yeast as well. That's why I asked Gözde this question. And this is our control place, and you can see at week 7 with the transform plant with GRF. The GRF technology and plus our transformation system allow us to extend the transformable genotypes more. Now we can transform Paragon and Cadenza, which we have got really good genomic resources with. Also, we know we can transform some elite commercial wheat varieties like Skyfall and Redlink. They are the recommended list in UK. And our efficiency is again very good. You can see the Redlink, you can transform 40% now. Previously, we thought we used the model plants for wheat, but in last meeting, we just thought we start to get a model plant now. We get a real genomic resources. We can use Paragon, Kronos, and Cadenza genomic resources directly, and we can transform the directly plant. So we don't need to use the model plant now. Unlike the other morphologic genes like the baby boom and visual system, we haven't got any detrimental effect in our plants, especially in low copy numbers. But we noticed that if the copy number is higher, GRF expression higher, there is some undesired phenotypes which can confuse with our gene function. And some of its varieties are more sensitive than the others, like Kronos. So we start to develop other systems, we can control our GRF system. Now Mark working on it and prepare some construct for us, and we start to test them. First one, we use is Krylog system. With Krylog combination with the heat shock promoter, when with increased temperature, we are just in later stage, it allows us to remove GRF in later stage in the transgenic plants. The other one is the controlling the GRF. We use the PLTP. It is the endosperm embryo-specific promoter, and it doesn't express in later stage. So we can produce plants, we can produce callus, somatic embryogenesis, but and later stage, it doesn't express the GRF. The other third system Mark prepared is the transformation. We aim to use 2 separate tDNAs. It means it's going to allow us to segregate away in later stage, and also we can use the previous construct. We just use it for filter, so it doesn't contain any GRF. So we can just use them together now, so we don't need to prepare new constructs. Third factor, I think one of the most important, also time-consuming, was screening, especially for polyploid species, because usually we just PCR them, amplify the gene, and after that send them the single sequencing. But like wheat, each individual subgenome needs to be screened for EDIS. This requires at least 3 PCR reactions and 3 sequence reaction per plant. It is a bit time consuming and you need to optimize your PCR. So we start to use now next generation sequencing, especially if we are targeting to 3 subgenome. This was our first published method, and it was targeted at only 5p copy in wheat. But it is actually a bit challenging because targeting, if we had a choice of 3 guys only, so it takes our time. So, but we were able to obtain a big plans with the large deletions, which contains 114 base pair deletion. Because the deletion was large enough, we just screened them with PCR and you can see the large deletions. This is homozygous EDIS plants and these are heterozygous plants and this is the wild type genotype. When we sent the Sanger sequencing, because it is just in one subgenome, you can see the large deletion by sequencing. Also, it is with the Cristobal and Cristobal's team, we tried, in this target, we just targeted a genome. So we just directly sent them, we PCR and sent them the Sanger sequencing again. This is again Cristobal and Cristobal's PhD students target, and in this one, they were targeted promoter region and they used the 3 guys, and they didn't check any synapse and small deletions. Just their target was large deletions. When that's why they only PCR'd. And you can see they got the 6% in Fielder and 7% in Kronos. They get some large deletions, and they even sent the Sanger sequence for it. And when we start to check the next generation T1s, it is most interesting because it shows us the deletions, actually mutations, carry on the next generation with CRISPR. The plants haven't got any deletions or mutations in first generation. They got deletions in T1. As you can see, it is 19% and Kronos is 9%, or almost around 10%. So it was very interesting for me to see the next generation, you can get more deletions. This is again with our work with Graham. He is the design feature with strategic leader. And this is our work with the collaboration with the Danforth Center in the United States. In this target, we were targeting the 3 subgenomes. And you can see we got homozygous EDIS in the 3 subgenomes successfully. The previous sample was in Fielder, and after that we tried something in Kronos, and you can see again homozygous EDIS in the 2 subgenome successfully again. Now, we are working on another system as well, because we always use the transformation system and tissue culture. But this method, high edit protocol, is allow us to produce edited lines. We may haploid edit with target genes. Wheat and maize, you can see that it fertilized after maize chromosome elimination, we can get homozygous edit without any transgene in it. This is our most recent detailed information and CRISPR system. Both our papers are open access and complement each other, and we have videos with them. Also, versions of other transformation method, you can see the with training, it's arranged by Cristobal's and Cristobal's team. And all the vectors we use, you can find in etching. I would like to thank the people we work together. I would like to thank the crop transformation and genome editing platform, but especially my team and Mark for the wheat transformation. We are the new team, but our results are very good now, very good at the moment. Also, I would like to thank Cristobal's and Graham's group. And also UC Davis, our collaborators, they are Juan and Jorge, they are amazing people, and they are open to work, and all the researchers at John Innes Center. Thank you.
Thank you very much for this great talk. Now, we can take some questions from the audience, please. If you can talk right in front of the can you come over here so people can see?
Okay. First of all, thank you for such an interesting presentation, and also Dr. Mark's presentation was so interesting for me. You say wheat sensitive than other crops. Why you think that? It's my first question. And I am a physiologist, and it's interesting for me. Equal to these changes at the molecular or genomic stages in the wheat. Did you investigate biochemical or physiological changes in the wheat plants?
I answered, I just like to explain.
Okay.
I think we can say a recursive secret. That is why it is the tissue culture response is very slow and very difficult. Only both White and Fielder have some good tissue culture response. That's why maybe first transformation start with varieties. What we always consider one of the most difficult crop to transform, and it was really because when I started, it takes our time to optimize all the protocol. But the GRF system make really revolutionary change in the wheat transformation for us. And usually we are working and collaborate with other people, but physiologic, some different targets they offer us to work on it. But Mark, can you repeat the second question, please?
Did you investigate physiological or biochemical changes at your wheat
Transformation.
Yes, transformation plants?
Actually, we don't check. We just looking all the full parameters, how our transformation affect the phenotype and other factor, but we haven't checked the phenotype stuff. Mark, have you got any? Because usually people working about like iron or starch, and they just approach us. They got the target gene, and we just blast find the sequence, and we design our guys or think about which strategy we can use for them. So we produce transgenic plants, check the copy number analysis for them, so they can compare their results, and they give DNA sample and plants them to carry on their research. So each again depend on our target and which people can approach us. If they ask us to carry on their more research, we can carry on it for them.
We can take one more question. So, can you come here because the audience online they cannot hear.
Okay. Yes, I understand. Thank you very much, Dr. Sadi, for wonderful talk, and I'm very excited. So, can you give a kind of a timeline from the design of guide RNA up to the first generation plant? How much time it is taking now?
Usually it takes 11 weeks from beginning to finish the transformation, but it's just only tissue culture stage until we transform them to soil. But with GRF technology, it getting shorter now. But we can say the design build guides, because a lot of them has to be transformation. So we need to just prioritize the works. But usually it takes 1 year. We design the guides, put in the wheat and transfer them soil, DNA extraction, copy number, and start to check the edits. We can check the edits for other people or people if they want, they can put this stuff.
So
but
probably
the more challenging part is the clinic now at the moment. But
but I think that probably is maybe
we can say 1 year.
1 year or even less.
it won't be as flat because now some people just approach us and give out the target and they ask us, our construct is ready, and we say, yes, your plans are ready now. Because with the GRF system, its transformation procedure is very shortened and we don't need to repeat anything.
This is amazing progress. I think that, let's congratulate John and his team, and we hope that, we'll be working together with all this. We were able to, at our institute, we were able to produce, transform all the varieties, but your efficiency is really good. Thank you.
Thank you.
We have one more question. Allah.
Thank you so much. Nice talking. Wheat is a polyploidy crop. How much challenging it is to generate medicated plants from such a high polyploidy crop? And for example, if you want to generate a knockout and getting a knockout with 6 alleles knocked out, it's challenging. How much challenging you find?
Now, I think it's getting easier and it's for us a routine because we are doing a lot of demand CRISPR. CRISPR is now, I don't know, everybody very popular and everybody interested in CRISPR. So we are doing CRISPR work a lot at Journal Center and it is our routine work. So most of them we just targeted 3 subgenome because you can, otherwise it's mask and you can see the phenotype. For us, it's routine process. If we can see the T0, mostly we can catch T1 homozygous edit in 3 subgene.
Do you often get a kind of a situation where some of the alleles are of the same genes are edited while others not? Because of some allelic diversity and all those things?
When I checked them, the next generation screening, it was in T1s I checked. You can see the A, B, and A, B, D, or D. So it is nice to comparison, especially it's important for gene.
All right. Okay, thank you.
Do we have any other questions from the physical participants? Otherwise, let me check the chat box. Okay, seems that we don't have any questions from the online audience. Thank you very much, Dr. Sadi, for joining us. Thank you. Thank you. We appreciate it. Now we will give 15 minutes of break, and right after that we will join again one more time.
From IFS, and several people asked that what can be done and what are the how we can keep this group going in the future. So I've especially requested Dr. Ismail to give his comment, and I'm sure that with his support and the support of all our colleagues, there's a lot of enthusiasm and a lot of progress made in the last 2.5 years when we met in Islamabad in 2019. And congratulations to Dr. Emre for arranging this meeting. With these words, I request Dr. Ismail to speak. And especially we think that by having some common understanding and coming up a program, especially on wheat, will be a great service to the whole humanity and also OIC member state. Thank you.
Thank you. Good morning. Thank you very much for everyone that gave me the thank you. Well, I'm actually We are running 16 programs, and they are all tackling the food security. And we are interested always to cooperate with our member states. OIC has 57 member countries. 36 of them are already member of IFS. So we are not only working with 36 member countries, but all OIC member countries. We work in strategic commodities, which is wheat, rice, cassava, and palm oil. And we are interested also for management water in agriculture. And how can we conserve our genetic resources? I think this is most likely in science. Among the other 16 programs, it is in IFS. We are not actually a funding agent, more we are cooperating with each other. We work for our member countries, and if we work for wheat, for instance, we could not work alone. We are not a specialist in wheat, but we have to invite a high caliber from our member countries or even outside member countries in collaboration ideas to write what we are today. Actually, we are not involved today. It is COMCEC-COMSTECH and Middle East Technological University. That it is, I came today also, thanks for the invitation, for inviting me to come, and also to start the channel and the bridges of linkage with the eminent scientists in this field. With COMSTECH, we will do a lot of activities during the 2022 and about action plan. And we are all talking about science. And we also cooperate with SMIC for halal food and halal and food safety. And we are cooperating with CISRIC. It's also for databases and food balance databases. So we can, our activities, we are thinking now to continue our activities. We have a new vision. The new vision will not go to concentrate only on those core commodities, but also we will make it a wider vision, because we have called from other member countries, very important, about date palm, about olive, and about potato, and about maize. IFS could not work in all of this alone, but we can work and coordinate with universities, with research institutes, with other OIC relevant institutions, that we can make this possible. So we are welcome that if you can, like the meeting today, or the training workshop, this one, we can make it next year in collaboration, for instance, with collaboration with COMSTECH and the university in another member countries, maybe in Pakistan, maybe in Kazakhstan. So we have to make this kind of activities. You can also at any time contact me, because we always need high caliber of scientists that to participate with us and to make our activities possible. We in IFS need you more than you need us. We are always tackling, and please also you can send me an email at any time. We published a newsletter about food security hub. We send it to the ministries of agriculture. We send it to head of universities in 57 member countries. So it will be very important to give promotion of your science or what you are doing for. It's not scientific review journal. It is a bulletin that we tell the people what we are in our member countries standing for the food security. And I think if you talk about food, wheat and rice, it will be very important and high value. So please, if you have any comment, any question, I'm here for the next 1 hour, and I'm here now because I do not want to take more of your valuable time, I know. So
It's a pleasure.
thank you very much. Thank you.
Thank you very much, Dr. Ismail. So his contact will be available to all participants, and Dr. Andre will share with everybody. Thank you very much for your time. And I think that I see amazing progress in the last 2.5 years from the time we met in Islamabad. So very pleased to see all this happening. Thank you.
Okay, thank you very much. Now we have the next speaker. In our program, our next speaker, Dr. Allah Baksh, is coming from CMB in the Center of Excellence in Molecular Biology, right? He has been working on transformation, construct development, and recently genome editing in plants. I know him that he was working in potato transformation for the last 6-7 years. And before that, he worked on many other crop species. So today, I would like to invite him to give a talk. This will be an example to all these previous talks, basically on the development of genome-edited potato lines. Thank you very much, Doctor.
Thank you for the introduction, but this may not be the example for others. Okay.
Now we need to do this. And then
How many participants can see it?
now they will see 1 second. They will see the share it. We will make it full screen. Okay.
So yeah, laser. Laser.
I don't think.
Is there laser? Yeah, perfect.
Oops. Oh my. It's frozen.
Mhm. Yeah.
Pointer C.
Thank you, Emre, for nice introduction. I put first slide for those whom I haven't met during this event. So basically, we come from University of Punjab. It's located right in Lahore, in the center. So the university was established back in 1882, when British were ruling this part of the world. So my Center of Excellence, is a part of this university and funded by Higher Education Commission Islamabad. My director, the next speaker, he will introduce with you that center. The center has different sections, and that deals with forensic sciences, stem cell research, some bioinformatics groups, therapeutic groups. So I belong to plant molecular biology. Plant molecular biology is one of a bigger group of the center working on different areas. If I say that mainly what we do, we try to introduce traits of economic importance in croplands. Cotton is the main crop, and among the traits insect and herbicide resistance is the one that the cotton that already been commercialized, and it's in the country for general cultivation for last couple of years. So this is what I can say that the center has done extremely good as far as the applied side of molecular biology is concerned. So besides that, projects have been conducted, are going on virus resistance. One group is working on improvement of seeds in different crops. And one group, we produce different events of as a result of transgenics. And one group is working on risk assessment studies of all those transgenic crops. And the next speaker, director, he's in charge of that group. So one group is working on edible vaccines for poultry. Basically, one of the group, their purpose is to improve the physiological traits in plants, especially in cotton. And why I'm saying cotton? Cotton is the major crop and the soul of textile industry in Pakistan. So we are also working for the fiber improvement of this crop. And to address all these challenges, we are using molecular tools like everyone. And recently, previously we were using all these overexpression approaches, then the RNAi, and recently the genome editing techniques. And among genome editing techniques, the talk of the town is CRISPR-Cas, and we started working with that. But here in today's presentation, I'll present some of my work in Turkey, because I was working in a Turkish university in Central Anatolia for the last 7 years. And some of the research I will present from this center that was not conducted under my supervision, of course, but my colleagues. But the two examples will be from our Turkish group, and that may not be a complete examples, kind of the research work that is still going on. Potatoes, the importance of potato is just like crystal clear. No need to explain about its importance. It's a hidden treasure. And we say that because we were in central Anatolia of Turkey, that is the best for potato production. And there we started working. And likewise, potato is also very strategically important for Pakistan as well, and of course for the world. And whenever we talk about the production, Turkey and Pakistan, they're almost at the same page as far as the production is concerned. And how important the potato crop is for the food security. And also when we talk about the climate change, potato again has a very important role in that. And as far as whenever we talk about the nutritional security, potato is one of the crop that can address the challenges. And this is what the journal is believed. And this is the report that can tell what kind of nutrients you can find. Because we were working in potato, so we developed transformation system. Like in a lecture before me, Dr. Sadia, she was talking about wheat, so one of the monocot. When we talk about wheat and potato transformation, definitely potato is a little bit friendly compared to wheat. So, it's we developed a transformation system using a reporter gene, the GUS gene, that was interrupted by an intronic sequence, and we transformed with agrobacterium, and we use a combination of different plant growth regulators, and we were able to establish a good transformation protocol in to make potato. And but using this protocol, we were able to conduct a couple of projects of TÜBİTAK in Turkey, and the same protocol is going on in my current institute. So this is one of the example in our current institute. Anila, she is a PhD student, also the research officer there. And she is trying to knock down this vehicular invertase gene. And the function of these genes is well established in Scrooge's partitioning, and also we percent of the edited region was found as so I'll keep on moving. Yesterday, there was a question, how do you select your vectors? And this is the beauty of recombinant DNA technology. You can design your own vectors, and there is a lot of talk of the town about the choice of promoters, choice of terminators, the people's public concerns about the GMOs. Keeping in view all those concerns, you can make your life easy by selecting different promoters, different terminators with your gene of interest. And also there is how we should drive Cas9 with different promoters. One of my students, he conducted this work, and Ilham Raham Kolov, right now he's a PhD student. This was his master work. And we just tried to drive this Cas9 with different promoters. And the promoters, what was the workflow that was, first, again, the concern, among concerns, one of the concerns is about plant selectable marker. And among plant selectable marker, we try to remove hygromycin, replace hygromycin with the kanamycin as a first step. Then we change the terminator and we put the plant-based, tobacco-based extensor terminator that is good in polyadenylation and is efficient in termination and transit expression. And then we try different, we clone different promoters with Cas9, and then clone sgRNA and did some experiments with ICRs. Okay, so I'm sorry, I'll quickly go through these slides. We removed, like I said, the hygromycin and replaced it with NPT2 because hygromycin is a kind of, there are some reports of regeneration problems and also some concerns. So we replaced with NPT2 first, and then we isolated this extension promoter from terminator, sorry, from tobacco.
That's a smaller one. And we just clone it in this intermediate vector, PGMTZ, and sequenced 8. Yeah, the same thing, we just cloned. Then different promoters, 35S enhanced promoter, and PAO promoter, YAO promoter was also used, and ubiquitin 10 and ubiquitin 7 and intron. Interestingly, we could not get result with this ubiquitin 17 or intron. I think we were not successful in cloning these promoters, but the other one was okay. These are the source of this YAO and this ubiquitin 10 promoter was Arabidopsis that was amplified, and there were 1 kb and 650 base pair. And then we cloned just first, like before, in intermediate vector and then into the final expression vector. This is the sequence, just confirmation. And again, with the ubiquitin 10, this is its confirmation. And these ones, which I said that we, my student, he isolated from this potato, but we could not get good results. Well, you can say we were not successful in getting those, but the 35S, yes, fine. It was quite fine with 35S. And then the next, we inserted Cas9 just upstream to promoters. So this experiment just shows that we were just trying to see the efficiency of this extension terminator, whether it works or not. So we used it with Gus and with different promoter, of course, and the terminator as this extension one. And yes, the agroinfiltration results, they showed that it really works. So Gus fluorimetric assays, they also showed that, yeah, in this one, 2 double enhanced Gus extension terminator, that showed the maximum Gus expression. And then guide RNA was, and this guide RNA is, of course, HPT, hygromycin phosphotransferase, the stretch. That was, the Gus was interrupted with this stretch, and guide RNA was designed against this stretch. And then the sequence was confirmed. Sorry for. Okay, and then intrachromosomal recombination assay, and the guide RNA the targeted this part, and then the Gus that got rejoined. We could see in our seedling. So this is was one of the students who will continue the same work.
Internet is fault.
Okay, my apologies to online participants. We are having some trouble here. Okay, so this cell wall invertase enzymatic activity is established that it's associated with pathogen infection in crops,
And invertase inhibitor genes can have direct or indirect role in plant host defense mechanisms. So the one we were targeting, that was apoplastic invertase inhibitor, and this is just literature that shows that how this invertase inhibitor is involved in inhibiting the activity of cell wall invertase, and how it's suppressing its defense mechanisms. And again, some results, I think I put a table just to understand more. Yeah, for example, here you can see some altered invertase activity. They reported in beet severe curly top virus, and in impaired defense mechanism was a result was found in when they silenced this this cell wall invertase in tobacco, and again Xanthomonas oryza against Xanthomonas campestre. So the point I just want to raise that this invertase activity upregulated in Arabidopsis essential in modulating carbon partition and nourishment of pathogens, and the increased level of this one will lead to the ability of plant to resist the pathogens. And the inhibitor is what inhibitor does, it just inhibits the function of that vacuolar invertase. So we used a diploid and tetraploid potato for that purpose. And yes, first we characterized the gene. It's a different exon which we were going to target. The exon one is the generally the one which is targeted in literature. The first one, so we first cloned and characterized and learned its sequence, although its sequence was already available, just to confirm in our the diploid and tetraploid cultivars that were under study. And we found that, but with the diet, they already reported that there are different alleles of this invertase inhibitor. So once the sequence was confirmed, we designed guide RNAs, and we cloned them in our target vector, but all this normal cloning procedures. The point I just want to make it is just we confirm sequences again. Then finally the expression vector were transferred to agrobacterium. The point I just quickly want to make here, which we faced in our studies. Maybe the people who are working on genome editing, they did not face. When we were trying to clone these our CRISPR constructs in E. coli, transfer our vector in E. coli, we could not get stable clones whenever we used JM109, DH5 alpha. So what resulted? We have to go for the XL10 gold strain that maintains the stability of these your constructs. So we were only able to get our clones in this one. And also we were really new. What we were trying to adopt a conventional cloning procedure, and always we were incubating our plates at 37. But what we learned, yes, by lowering the temperature at 28 and 30, you will get better colonies and more correct clones. And at 37, we were getting 1 or 2 or sometimes no colonies. So these are different explanation what can go wrong with this vector stability analysis. This is what we observed. Maybe the one people may not encounter with these problems. Then these are some results from from the calli. Some of the calli we just tested our constructs, knockouts in calli, and we cloned. Actually, these are the results of cloning, and these sequence shows the insertions, deletions, sorry. But interestingly, we also could not find our the gene edited one when the plants grew to a larger size. In some of the plants we could not find, but I mean at callus stage, yes, knockout was there, but when plants grew to a certain size and we checked again, the knockouts could not be confirmed. This is what also we encountered. And what we saw that, okay, also the Dr. Gozde, she also talked about, we tried different temperatures. And the 37 for the Cas9 to work efficiently, and 37 with the different and co-cultivation at 28 for 2 days, and we transfer intermittently to 37 while on Kala induction medium. This was also good to get results, because we kept on changing temperature and giving at 37 centigrade, because the keeping your cultures for 37 for a couple of hours times overnight may enhance the activity of Cas9. So these are different related stuff related to Cas activity. So the main point is that plants, we just check them, Xeria and M6, this is deployed cultivated variety and the control, and also some of the Xeria lines compared to the control going on. And many more experiments are needed to confirm and establish the genome editing in these potato plants. Had examples for the moment, and I would like to acknowledge, of course, my previous university, Nido Morales Demer University, for providing us a good platform for the last 7 years to carry out our research work. And then my Center of Excellence in Molecular in Lahore, where I just recently shifted. And then Erasmus, and in Erasmus, during Erasmus time, my student had a good work in James Hutton Institute with Dr. Abdullah Barakat. I'm really thankful, Dr. Emre Aksai, in all this CRISPR-Cas work. He was the very active member. Professor Memtmin Chalishkan is a breeder, potato breeder, and always he was helpful in providing us the materials, and also teaching us how to keep this material grown in greenhouse and in field. So in camp, Abdul Qayyum Rao, who is the PI of the work I mentioned about vacuolar invertase, and Professor Dr. Ahmad Ali Shahid Sahab, who is with us here and will present next lecture. With this, again, by taking advantage, I really want to thank Dr. Emre Aksai and Dr. Shahid Mansoor for inviting us. It was great to be with you and to learn and discuss and interact with you all. Thank you so much.
Okay, thank you very much for this great presentation. Now we have 20 minutes to take a few questions from the physical participants. Are there any questions from the audience? Yes, can you come here? It will be great.
Thank you very much for a very nice presentation. My question is that, one of the target gene other than vacuolar invertase. So you were saying that, in your slide, that probably you need to overexpress to have disease resistance. But then in your work, you were trying to knock it out.
Yeah. This one is cell wall invertase inhibitor. So this actually what happens, this inhibits post-translationally, this inhibits the activity of cell wall invertase. So what we wanted, we just wanted to knock down invertase inhibitor in order to increase the accumulated accumulated level of cell wall invertase, and the increased level of cell wall invertase means more resistance to apoptosis.
So have you confirmed the phenotype?
No, this is what is left. This is what is left.
Thank you very much.
Thank you.
You may ask for online.
Now we will look into the online participants. If there are any questions in the chat. Just see that. Okay, it looks like there are no questions in the chat. If there are any questions from the online audience, we can take them now. Okay, seems that I think there are no other questions online as well. Thank you very much, Doctor, for this great presentation. So, we can finish in 25 minutes. Okay. So basically, since we have finished this presentation slightly earlier than our program, therefore we would like to continue with our next speaker, and this will end our presentation session actually. Then we can give a lunch break. So our next speaker, Professor Ahmed Ali Shahid, is also coming from CMB, Center of Excellence in Molecular Biology in Pakistan. Dr. Shahid is the director of the center, and he is working on the regulations and public acceptance of transgenic plants. So today, I would like to invite Professor Shahid to talk about the regulation and public acceptance of genome-edited crops in the world. Yes. I will just prepare it. Share the screen. This is the pointers. Okay.
You can it's full screen there.
If we make it full screen, then actually we lose everything. So you can just see it by using these buttons. It moves.
Okay.
And we see it actually full screen here.
Okay. Bismillah ar-Rahman ar-Rahim. First of all, I thank for the opportunity to present something about the regulation and public acceptance of genome-edited crops. So I am working as a professor in this Center of Excellence in 1984. So we extend this center. Before going to, I will tell some which are techniques which are being used for the modification of crops. So first is the crossbreeding. Crossbreeding, combining two sexual compatible species to create a variety with the traits of the parents. Then the mutagenesis, use of mutagens such as radioactivity to induce random mutations creating the desired traits. Then the
ID is the multiplication of the number of chromosomes in a cow to impact its fertility. The next technique is being used as the protoplast fusion of cells or the components to the transfer traits between the species. Then the next technique is transgenesis, addition of the genes from a species to create a new variety with desired traits. Then the genome editing is the techniques use of enzyme system to modify DNA directly with the core, with the cells. So then we focus in this conference on the gene editing. So how CRISPR works, is a general view. So it has the 4 steps. First is targeting. First we select the target. Then the binding process. Next is the cleaving process. And the final process is DNA repair. So in targeting, the scientists introduce the Cas9 guide RNA complete into a cell where it's randomly associates and dissociate with the DNA. Cas9 recognizes and binds to a 3 nucleotides sequence motifs called ramp that is abundant throughout the genome. This is the first step. Then the next step is binding. In the binding, once it binds to a PAM motifs, Cas9 unwinds the DNA double helix. If the DNA at the location perfectly matches a sequence of about 20 nucleotides with the guide RNA, the DNA and the matching RNA will bind through complementary base pairings. The next step is cleaving. In the cleaving, DNA RNA pairing triggers the Cas9 to change its 3-dimensional structures and activates its nuclease activity. Cas9 cleaves both DNA stands at a site upstream of the PAM. In the cleaving also, the DNA RNA pairing triggers Cas9 and the changes its 3-dimensional structures again. Then the next step is repair. So in repair, it covers two steps. First is non-homologous ending joins, the other is homologous ending joins. A non-homologous ending join is more frequently used, faster repair mechanism because the cell does not use a template to join the broken DNA, and together it is however an error-prone process that can introduce mutation in the target sequences. If the target sequence is within a gene's coding region, the mutation will likely inactivate the gene. In homologous directed repairs, the second type of repair mechanism is homology directed repairs, which is a less error-prone and uses a homologous DNA template to accurately repair the break. The scientists can manipulate this repair system by introducing the cell and access of a DNA repair template along the Cas9 guide RNA complex. Advantages of this technology. Offsite mutation can be avoided. Any gene can be targeted. In this technology, we can genes. A row of improved traits have been introduced in different crops already. So there are so many crops which are being targeted by this technology, and they are giving good results, and they're improving their qualities and their production also. Now the CRISPR case application, the selection of the targets. Yesterday there was a few presentations on this technique. So then after the selection, then the designing and synthesis of the sgRNA and promoters in plant binary expression vectors. The next step is the deliver vector to the plants. Then it has two steps. By the two steps, first is the agrobacterium transformation, then in the particle bombardment. So by using these techniques, which is available in your resources, you can use any technique to adopt this technology. Then we have the putative plants. The next step is screening of the putative transgenic plants. It also contains two steps. First is the PCR and restoration enzyme analysis. The other is the DNA, the plant that contain the next generation advanced that contain the desired for that. So many importance. So the first is it effect on the agronomic values. So we can see in the wheat by using this technology, the wheat can be obtained by in the bigger grains that also increase the grain weight. If the grains are bigger, also it is automatically the weight will be increased. By this technique, the only female flowers can be produced to increase its yields. Then in canola, that increase shatter resistance, because this shattering is very, due to shattering there is big loss in the crops, so they can be increase shattering resistance. Then it also be useful in food and feed qualities. In the soybean, it can be obtained the improved oil quality, and in rice, reduce the arsenic contents by using the technology. And in wheat, which is very important crop, then reduce the gluten content by using this technology. It can also be helpful in stress tolerance in the grapefruit and orange. In rice, wheat, and tomato, this can be used to control the poddy mildew resistance. Then in grapevine, it can also be useful. Then it can also be useful in the maize, wheat against the drought tolerant, and in rice this can be used and the salt tolerance, and in soybean it can be helpful drought and salt tolerance. Then it's also be increase the fiber quality of the wheat, and also drought tolerant maize can also be produced, and also in the potato with reduced PPO and glycolides contents. Then in wheat also mildew resistance wheat can also be produced by using this technology. Then it is also helpful in the food security, and then climate crisis it can be used helpful. Then biodiversity conservations, and then the dependence on the heavy inputs and fertilizers, pesticides, and irrigations, we can minimize these things. Then there are four techniques which we can use. We can edit the gene. The first is the ZNFS, then the TALENs, then MN, and then the CRISPR. So this techniques has the four categories, and the first category is SDN1, and the second category is SDN2, and third category is the SND3. So in SN1, SDN1 application mutation consisting of the changes in a few base pairs, and the short deletion or insertions are generated in the finding regions in the genome as a result of the error-prone gene repair mechanisms of the cell. In SDN2 application, specific point mutations, small deletion additions are generated as a result of the introduction into the cell of the repaired DNA templates. In SDN3 applications, entire genes can be inserted in the desired site in the genome. Then the process versus product-based GMO regulatory frameworks. So some people believe on process-based technology and other believe on the product-based technology. In process-based, they consider the techniques used to create new crops, plant varieties. So they take inquiry about which process was used for these techniques, then regulated if nucleotide acids are deployed during the development of a crop. And in product-based, they focused on the final product, no matter how it was developed, based on the potential risk of the new trait or attributes introducing into the plant to a public or environment. So this is the slide showing the landscape of the regulations. So there are different countries are adopting different techniques. Then in Canada is considering the product-based technology, and in the USA they are also opinion about the product-based technology. Then in other Colombia and Brazil, they are product-based technology. They believe on that. And other countries like European Union, they believe on the process-based technology. And Russia still to decide. They did not decide. Still, they are thinking about that which technology they should adopt. And Australia, they believe on the process-based technology. In New Zealand, they also process-based technology, they are adopting this. This slide shows the NBT assessment roadmap. So there are a few steps in that. First is the biosafety committees. Then there a new combination of the genetic material. So if they say no, and other side, then they have, does the NBT use a transgene temporarily? Then if they say yes, then that is a GMO. And then next is does the NBT use a transgene technology? If yes, then is the final product free of the transgenesis? Then they do more things about that. And if they say no, then they say GMO. If they said yes, then they go to the So there are different countries showing Argentina, Brazil, Chile.
Okay, we can continue.
So now we will talk about USA. The USA does not regulate or have any plans to regulate plants which could otherwise have been developed through the traditional breeding techniques, as long as they are not plant pest or developed using the plant pests. So they have the website MI regulated under 7 CFR parts 340. Then, but deregulated organisms by the USDA APHIS may still be subject to other regulatory authorities. In Israel, Ministry of Agriculture and Rural Development, National Committee for Transgenic Plants, then the gene editing, there are some that the Commission reconfirms its previous decision dated that plants which are the progeny of plants that underwent targeted mutagenesis utilizing gene editing methodology that caused the deletion of nucleotides and were proven that there was no insertion or incorporation of the foreign DNA into the genome of the plants do not fall in the category of transgenic plants. Therefore, plants, their products of this technology will not be considered transgenic. In Australia, new decision 10 April in charge, then the no regulation for the use of gene editing techniques in plants, animals, and human cell lines. They do not introduce new genetic materials. Valid for the use of tools in which protein cut DNA at specific target sites, as long as the tools allow the host cell to repair the break naturally rather than using a template containing genetic material to direct the repair process. In Russia, Russia is embarrassing gene editing. Federal programs wants to produce 30 gene-edited crops and animals by 2027. Some gene-edited products will be exempted from a law passed in 2016 prohibiting the cultivation of the genetically modified organism in Russia. New decree describes gene editing technologies which do not necessarily insert foreign DNA as equivalent to the conventional breeding method. In Europe, then old GMO regulation, but discussions are ongoing. EC takes a new attempt to assess NPBTs by April 2001 to start a science-based decision process already started. So they define the GMO means an organism in which the genetic material has been altered in a way that does not occur naturally by mating or natural recombinations. So this slide shows the current status of EU legislation on crop breeding. So we can see here, though the mutagenesis, they consider it safe, although it has so many things which in the mutagenesis takes place and there are so many steps. And also in the crosses, there are also mutagenesis, other ways technology, but they considered that it is safe. But in CRISPR case technology, where is only gene editing is used, so they are thinking that it is dangerous and they are very reluctant to adopt the technology. So we have to think what are the behind with these things, why they don't accept this technology. Well, actually, there are so many differences. The other technology are very complicated, but this is very simple technology, or they will not accept these things. Then these are the policy developments in different countries around the world. So we can see from this slide that the green things is positive decisions in these countries. they are taking the positive decisions. They are thinking to accept this technology. And then in the yellow, there is a no farmer decision on these countries. And in the red, whereas red is showing that is the restriction decision. So we can see that in Canada, product-based approach, ongoing discussion to improve the systems. So they believe on product approach, but there are always chances to improve this technology. Then in US, proposed new USDA policy excludes certain techniques, executive order to modernizing the regulatory framework for the biotechnology. In Argentina and Chile, Brazil, so they case by case the certain in your the decision cells mutagenesis GMO and the subject at EU GMO regulations. In Russia, we can see that they declares that conventional like as GMO. In China, proposed. In Israel, outside the GMO scopes. In Australia, revised gene technology. And in the New Zealand High Court decision that few specific techniques are GMO. In our country, there is also government has their case by case. But they will first they check the case on which crop and which technique is used. Then they will decide can they adopt it or not. Then this slide shows the study presenting experimental field trial data. So in this we can see in USA they are adopting the tomatoes. Then Sweden potato, and England also they are using cauliflower. In China tomato, and Japan they are also adopting to producing the tomatoes. So this is the last slide. This we can conclude from this presentation that publication on genome editing applications are increasing continuously worldwide. Then CRISPR's case is the dominating tool that is being used, and with passage of time, the people will be used to adopt this technology. And in the beginning, there are some problems, but ultimately the people will come to adopt this technology. Most applications that are 90% introduce targeted mutations. Only 10% insert heritables, that is foreign DNA. The China and USA are the leading countries to adopt this technology. Then application address a huge diversity of different characteristics, addressing all kinds of relevant agriculture or consumer traits. Then more and more countries follow a differentiated regulatory approach, taking into the account the kind of genetic change. And this is about the conclusion. Then at the end, I will acknowledge the scientists who are working in area of the genome editing for their continuous efforts towards food and nutritional security. Then all researchers who publish their works in peer-reviewed journals on genome editing. So thank you very much for your attention.
Thank you very much for the presentation. Do we have any questions from the audience? Can you come closer to the computer?
Thank you for your presentation. I'm sure that most of the people sit down here, that's what now going in the climate change is by handmade of the man. So my question, in natural, that's not happen that of maize can cross wheat, that's an example just. So foreign DNA in a certain crop, what's the effect or other consequence on the wild progenitor especially this wide originator will stand for a decade. So what the effect when we started to plant any crop and have a foreign DNA and plant it in the field. There's an study or expectation in the effect and the consequence in the wide originator that in fact this is the storage DNA and will stand for a decade and maybe losses maybe. But there's any expectation for this?
Yes, because we are playing with the nature, because nature has a set process. When we insert or delete any DNA or changes, we are expecting that there should be, we expect that maybe they will go beyond their natural nature process. So to get this technology, they have, though people are worried about that, maybe the basic thing may be changed on that.
But gene editing, it's not a nature.
Yes.
So maybe we lost the wild type originator. How can keep it for the next generation, not for our generation, for the next generation? How can keep it?
So we should continue practicing this, and we should closely check healthy steps. If there is any change, then we can use the technology. We can use it.
Can we control this? Can we control, in fact, in the world, can you control the distribution with field with full G and foreign DNA?
Yes, up to extent we can.
And not to extent, I sit here and we talk about in general, not in Jordan, not in Pakistan.
Yes.
Here taken in the future for a generation, not for us.
Yes.
So how can he save it?
So because it can be safe because the population is increasing day by day, and we have to adopt this technology. Otherwise, there will be no food. If there are so many diseases and there are so many threats to the class. So we have to accept this technology. Otherwise, we will have nothing to eat.
So you are saying destroy the future?
To save the future.
To save, no, that's not save the future. If you are lost the wild originator, right? Okay, thank you.
That's okay. Yes, that's it.
I think that there are many people that can answer the question, but just to be for general audience, obviously, this is a question that has asked many times to people like us. So there is a safe history of use of genetic engineering. We have been using transgenic cotton, maize, canola for a long time, and so there's a safe history of safe use. Now, whether to plant it in a country or not, that's a political decision. The government make a decision whether to. So there are countries that allow you to plant GMO, and countries that support either because of the trade or because of some other unknown fear. So I think that, but the history is very much there. Transgenic cotton for 20 years seen any adverse effect. Much safer because it's just like having is a dilemma that EU mitigations for creating mutation, and it is just like are happening because you select the best mutant. So this is something which is acceptable to the whole world, but genome editing is relatively, I will say, is considered as safer because in most of the cases there is no transgene. So most of the countries have adopted a very practical approach. I was very pleased with the very practical approach of Japanese and Australia. So I'm sure that there are gene banks available with thousands and thousands of lines of every crop species. If you are not interested to grow, and if you want to, remove one of the attitude crops, you can always do this. As in agriculture, we are replacing varieties very quickly. So especially in case of wheat, the varieties are replaced after every few years. So I mean, this is an ongoing process that you select the best, and if it is not working well, then you can replace it. So if there's no that the GMO will stay always, or edited crop will not, as long as you have some benefit, you can grow them, and then if you are not very happy with, for example, you can replace it with another variety. Remember the comments by Dr. Mark Tester. He said, I mean, the important is that we want to have food security. And genome editing is one of the technology. Dr. Mark and Dr. Haita has really very well explained that that how important it is that to mutate all the 3 genomes. It is something which is not feasible a few years ago. Is a remarkable development in the history of mankind that we are able to do very quick and very specific mutations. I must say that so this happens whenever you bring any new technology. Even there are people who are against vaccines. People who say we don't want our kids to be vaccinated. So this is always a choice. So this is the society as a whole decides that whether you want to adopt or not adopt our technology. We are here sitting here. This meeting, it was possible because all of us are vaccinated. So this is a remarkable achievement of mankind that within short time we were able to come up with vaccines, and so there are people even in the developed world that who have doubts and they don't want. So this is a free choice that you have, that whether you want or not. So I think that, sorry for a long comment, but I think that it's very important that we keep the science progressing, and adopt the whatever methods are available, and as a society decide whether they want to adopt or not. There are some of the I mean, when we were starting our biotechnology program, there was so many ideas that could not see the light because they were not. I mean, they remain an idea, but there are others who have gone into the field and created an impact. So so we'll see. So but we'll continue to make process and then make choices. Thank you. Sorry. Dr. Haida.
I think you give me, I can ask, because I don't know the regulations in Turkey and Pakistan. It is that now hosting countries to organize this workshop. So could you tell me what is the regulations in Turkey and Pakistan? And also second stuff, is there any strategy to promote the genome editing in Turkey and Pakistan, like outreach activities or our awareness to publicity, this kind of activities, there are activities in the countries?
Sure. I can tell about.
Thank you for explaining. This was great.
I can tell about Pakistan. So there's a regulatory authority. It's called National Biosafety Committee. There's a technical advisory committee. I co-chair that advisory committee in Pakistan. And in 2019 we held the meeting, and we, as a country, we welcome the technology, and we said that we will do. You still have to apply for biosafety clearance because as Dr. Ahmed Ali Shahid has told that there are 3 possibilities that you can also have a transgene when you are doing theoretically. This mostly it is knockdown. So the policy is that we will discuss the applications, and most likely we will declare whether the technology is going to lead to the in the final product, whether there's a transgene involved or not. If there's no transgene, if it's just a mutation, we usually be considered as non-GMO or freedom to operate. So this is the national policy of Pakistan, and we are promoting this technology. So maybe Emre can tell about the Turkey, but I'm sure that probably similar situation in Turkey.
Now, basically, this right now in Turkey, we are following the EU regulations. So, and we just need to see their decision on genetically modified gene-edited crops. But other than that, for the general GMOs, it is highly protected, regulated. We can produce them. We can establish the lines in the laboratory. We can take them to the controlled greenhouse conditions, but we cannot take them out. It is illegal. So all the seeds we import, they are highly regulated against this GMO. From the ministry, they may have some extra things, but not this much. If there are anything change.
A part of targeting for we sign the protocol. That's why we should follow the protocol. But now, for now, we are not allowed to any of trials. We just allow to use the GMO products for feed production, not feed production, just for feed usage. We import the seeds or some plants from. We are importing some seeds for just feed usage, not anything else, and also some for painting industry. I think that's all. We are not allowing as for now. And it is the same. There isn't any difference for the yeah. There isn't any difference.
We have some comment at the back. Yes, you can come.
Actually, we always talk about the sites that we always, in terms of consuming GMO products. What about only seed production? I mean, those countries, do they allow seed production in their own land, in their own countries? For example, European Union, there are many companies, they invest on GMO and gene editing. And at some point, they have to actually test this on some field, and then maybe under different environmental conditions, they want to select different lands, different countries. What is the actually legislation about this? For example, in Turkey and other countries that are restricted against, they restrict using GMO as
I will tell about Pakistan. In Pakistan, there is a if you have the transgenic or any GM crops, so you can grow in your country. There are different legislation bodies are there passing through them. There are some rules regulations. Then you can grow into your own country. But we cannot produce you export to other countries. But I don't know about Turkey and other countries because each country has its own regulations. So they adopt their own regulations. But if they export a country, there are international regulations. So they follow that regulations.
I can add some. So it also depends on the trade. For example, we allowed rights for maize in Pakistan. for 3 years, and then the case was finally good for the commercial application, commercialization. The field trial is allowed, the commercialization was discussed, but the grade that were offered were, insecticides and herbicides. And the overall feeling was that, these are not really the grades that are required by the farmers, because the situation is changing. In maize, so cotton is grown. 95% of cotton is produced by Pakistan. For maize, we said, okay, for the time being, there's not much benefit to the farmers. But with the rise of fall armyworm, we are thinking to reconsider cultivation of maize in Pakistan, because there's a big problem. So 2 years ago, there was no fall armyworm in the field. So we didn't see any benefit. And also, the other problem with the maize was that the pollen travels a long distance. It has so, but the thing depends ultimately on the trade and the benefit of the farmers. The farmers have the benefits are good. Thank you.
I just know one case for Turkey, and I remember late 90s or early 2000 in Turkey there were some transgenic corn trials in the Adana part, and then I have studied what I have seen in different conferences. People talk different about the results. Some people, actually the results were conducted under the Ministry of Agriculture. So some people show a data where the transgenic corn, the yield was almost double. And but some people don't believe those results. So it's a kind of a debate still going on. But yes, it was a foreign company that somehow conducted their trials in the country. But after then, there was, you can say, there was no any trial. This is what I know conducted in Turkey about the transgenic. And yes, this is the case. And the earlier question, I just want to add, I got an opportunity. The people who like genome editing who are hopeful. In EU, there is a consortium called SAGE. This is a consortium of 137 countries. And they're kind of pushing against this PC, this definition. And they very recently, Wendy, she was one of them. They published a very good article and defining how transgenic and how we can issue that we're not in EU or from ministry. If something changes in EU, definitely it will impact many other countries, including Turkey.
Also, I think UK has differently.
Because because we didn't now. The basic thing is we can do now our research is not depend on regulations. So now as we can do field trials, especially the genome editing process for research purposes. So it is great development for us. Also in UK there is a kind of promotion of genome editing kind of for public awareness or the publicity or outreach activities, and it works. Now farmers are more open to genome editing process, and they are able to use it, but they are just considering how they are going to find money. Because if they can sell their products in Europe, they can make money. That's the only problem. But obviously, I don't think UK wants to is the opportunity to use genome editing for breeding, but it's shortened period and can distinguish from the normal mutation period. With the mutation period, I told you, 500 has more mutation mutation, and we can't increase it, just upgrade.
and they don't want to use this opportunity because if we just do research, we are agriculture produce, but it can come to market, but from United States, so they don't want to use this opportunity. So we are open with that, but just we are ready to because for us, the market, and also now we can use it for research purposes, but eventually in future is going to be more open.
Thank you very much. Because I have some small comment. I have been working with transgenic plants a long time, and several transgenic plants, including rice and other processes. We observed unexpected metabolite changes. Not on all of them, but some of them. But especially transgenic plants generated by tDNA mutagenesis, sometimes made very hazardous. Some of them were observed very strong accumulation of thermal compounds under hot stress, which can be accumulated on the seeds and then they cause allergic reactions. So in that case, I think we should not go only for the new technologies. Also, we should improve all technologies in the midst of the making our products better, especially for food business. We should be very careful. Thank you very much.
Well, this is very interesting because in literature, if you look at different articles, except papers from Chavini group in France, there is no, concrete kind of evidence. I must want, if I want to introduce the word substantial equivalence. If your transgenic crop plant was not substantially equivalent with your control, yes, definitely you can say that there might be some pleiotropic effect of your introduced backbone genes. That's quite possible. But the beauty with the working with the plant is that you can discard your plant if the phenotype is different. You just discard, and there are regulatory mechanisms, frameworks. What we learned from this lecture, there is the main mandate of those regulatory frameworks if to assess all these genomic data, GMOs plants at every step. And once all the parameters are completed, then they are allowed for the next step. So all things are in consideration.
Yeah. We actually did a comparison of transgenics and non-transgenics and plant through tissue culture. And we actually found that there were more changes in plants which were produced through tissue culture. There were changes in metabolites than what were in the transgenic and transgenic plants. So tissue culture is quite a stressful environment for the plants to regenerate. So we actually found more changes through the tissue culture transgenics. So I think every transgenic plant or event needs to be assessed on its own areas, and it really depends on the position that the tDNA has gone into the plant, and also the gene that you're regulating down regulating. This kind of thing.
I just want to add something. I think the technique is not the problem, but the thing is the dynamic of genetics. We should know the dynamics of genetics and then how one gene affects the other, how one gene affects the other gene affects. So we have to think more, on a macro scale, and then we have to see the effect of that phenotype I'm sorry, the documentation, and then which phenotypes are affected simultaneously. So it's not actually related with just gene editing, the CRISPR methodology. Yeah, you can do this with some clonal mutation under the tissue culture environment. So it also happens in nature. Mutation. So you can do, you should be aware of your
Let me see. Okay, in the chat, we don't have any questions from the online participants. Now it is 12:30 in Turkey, and we will give 2 hours of break for lunch. And afterwards, we will come back for the online presentations of some posters. We will have, I think, around 14 posters. We'll have 5 minutes. And right after that, we will finish all the lectures. Thank you very much. Thank you. Thank you. Now we will start with the poster sessions. Now, in our list, the first speaker is Meher Hassan. So the title of the poster is Root Retention and Translocation Defines Cadmium Tolerance in Mung Bean. Dear Meher, can you share your screen so that we can see your poster? And please, you will have only 5 minutes to present your poster. Just keep with the presentation duration.
Where is the slide? Bismillahirrahmanirrahim. Assalamu alaikum. I'm Dr. Meher Hassan, an IPFP fellow from Department of Genetics, University of Karachi. I would like
we
to
can now
thank,
see your screen, so you can start.
I have already, you're sharing a screen, but I'm sharing a screen why it's not visible to you. Share.
You can go back to the original share, so I think that will look better. You don't need to make it full screen.
Okay, okay. Escape. Okay, now it's clear. Can you
Yes,
see
we
the
can
screen?
see it clearly. Yes.
Okay. Bismillahirrahmanirrahim. Assalamu alaikum. I'm Dr. Meher Hassan from Department of Genetics, University of Karachi. I would like to thank COMCEC and COMSTECH for providing me opportunity to present my work at this wonderful workshop. My topic entitled Root Retention and Translocation Defines Cadmium Tolerance in Mung Bean. First, I would like to introduce about cadmium and mung bean. As we know that as a sessile organisms, plants are usually exposed to so many abiotic stresses. Among them, one is heavy metal stress. Among heavy metals, cadmium is a non-essential metal that is responsible for significant growth reduction, ultimately leads to disturbed agriculture production. In Pakistan, almost 32 to 33,000 land is being irrigated with industrial effluents. Regarding my experimental material. Mung bean is an important leguminous crop grown throughout the world. Almost it's had been growing on the area of 250,000 hectare area and produces 180,000 tons per year. It is sensitive to cadmium, so it's a good bioindicator for cadmium contamination in the soil. There are different resources of heavy metals in soil. For example, there are industries, industrial effluents, transportation, mining, and use of different fertilizers causing contamination of soil due to cadmium. In our experiment, what we have done, we have taken different varieties of mung bean collected from NARC Islamabad, and these seeds were sterilized in bleach and then sown in Petri plates, transferred to Hoagland solutions in aerated plastic tanks. Then we have applied 0.1 micromolar cadmium to the plants. After this, after 1 month of the growing plants, we have done with the harvesting. We have noted morphological parameters, and then we have performed quantification of roots, cadmium in roots, shoots, and xylem through ICP-MS technique. Then stats was performed in order to know our results. Okay. In results, we have observed the growth performance was severely affected, as we can see in these pictures. These are the control plants. They are looking like healthy plants, while in treated plants, we have seen the chlorosis of the leaf. So it's the indication for cadmium toxicity in the plants. Regarding our morphological parameters, we have seen that among 8 different varieties, we have poor growth in NM51, and there is good growth in Azri variety. Regarding root and shoot length, we observe the same results. And if we talk about the cadmium amount in root and shoots, we can clearly observe that most of the cadmium is retained in roots, while minimal amount is transferred to shoots. Here we can see that in Azri, the growth is good, and in roots, we have the higher retention of cadmium. While here in shoots, we can see that both the sensitive varieties have higher amount of cadmium in shoots. Here, similar results were observed in cadmium translocation. Here, NM1919 and NM51 have higher cadmium translocation, while Azri have the lowest cadmium translocation towards shoot. So in conclusion, we can say that both among all varieties, growth parameters were decreased during cadmium stress. Higher tolerance index was observed in Azri, while lowest in NM51. Interestingly, NM1919 translocated higher cadmium, but with good vigor, so it can only be grown in cadmium contaminated areas to monitor cadmium, while it's not suitable for edible purpose. And here we can also say that plants translocation and root retention properties are good in defining cadmium tolerance ability in different crops. At last, I would like to acknowledge European Commission for providing me scholarship under Erasmus Mundus program to conduct this research in University of Milan, Italy, under the supervision of Professor Saki. Thank you so much for your attention.
Okay, thank you very much for your presentation. We can take one question if there are any. We can take questions from the online audience as well. Okay, I think we don't have any questions. Thank you, Meher, for this great presentation.
Thank you.
Now, we can continue with the next presenter, Kasım Raza. He will talk about Matchbox genes galore in wheat genome. Galore, genes galore.
Hello, can you see those slides?
Yes, we can see your slides.
Thank you very much for giving me the opportunity to present my work at one of the top scientific forums. My name is Qasim Raza, and I am an early career researcher at Rice Research Institute, College Shakapu, Pakistan. Bread wheat is one of the most produced, stored, and consumed food crop worldwide, and it is a major source of energy and nutrients in the developing countries. However, after green revolution, the improvement of wheat grain production has been hindered by various bottlenecks, especially due to the non-availability of a reliable and fully annotated reference genome. Recently, the IWGSC released a high-quality genome sequence of red wheat, and which is the largest known genome in the of plants. Alongside the reference genome, the large-scale RNA sequence data expression repositories, they provide a rich resource for wheat functional genomics, and which in turn could facilitate characterization of important gene families and crop improvement efforts. Medbox transcription factors, they are a well-documented group of genes known for playing multifarious roles in regulating the growth and development of crop plants, and they hold enormous potential for bolstering grain yield potential. The objectives of this study were genome-wide identification of Medbox genes and to study the gene family diversity through comparative phylogenetic analysis, as well as to investigate the potential mechanism for gene family expansion and evolution, and finally to explore their expression patterns under different abiotic stress conditions. We retrieved genomic CDS and protein data of all genes from the ensemble plant database and verified through NCBI concept domain database and smart databases. And then the multiple sequence alignments were generated with the MAFFT version 7 and a maximum likelihood phylogenetic tree generated with IQT software and visualized with Mega 7. We also checked the sequence similarity among the genes through sequence demarcation tool and considered those genes as duplicated, which showed more than or equal to 90% homology in their sequences. And finally, we retrieved the expression data of all these genes under 16 different abiotic and biotic stress conditions from the gene expression browser database. Through a combination of different search approaches, we identified a total of 300 high confidence and non-redacted full-length protein encoding Medbox genes. And to date, this is the second largest number after Brassica nippus, which contains 307 Medbox genes. And if we also consider the low confidence and incomplete protein encoding Medbox genes from wheat, then wheat has almost the largest number of Medbox genes. And these genes, they are almost equally distributed among the 21 wheat chromosomes, and with the exception of 3 homologous chromosomes on the 7th linkage group. And the percent contribution of the 3 subgenomes, they were also comparable. Furthermore, the comparative phylogenetic analysis among Arabidopsis, rice, and wheat Medbox genes, and they exposed 14 MIKC type subfamilies and 3 M type subfamilies. Interestingly, the previously annotated FLC-like genes, and they were grouped with the MIKC star-like genes. As you can see here, And furthermore, through comparative phylogenetic, comparative multiple sequence alignments, we demonstrated that the amino acid sequences of the Arabidopsis specific FLC-like genes, they were significantly different from the MIKC-like genes. And these results indicate that wheat and rice genomes, they might lack FLC-like genes. Moreover, we found that at least 8 subfamilies, they were significantly expanded during the gene family evolution, and a majority of the genes belonging to the significantly expanded subfamilies, they were located on distal chromosomal segments shown here in purple color. Remarkably, the duplicated gene pairs, they were subfamily specific and particularly recognized in subfamilies containing the unbalanced homologs. Unlike the MIKC type genes, the M type genes, they have not been extensively studied in crop plants. And in this study, we observed that M-type genes, they were predominantly located in distal telomeric segments, and they are evolving at a faster rate, probably due to higher frequency of tandem gene duplications and stronger purifying selections. And our results, they highlight the significance of M-type genes in understanding the evolutionary history of bread wheat. Finally, we examined the expression patterns of MADBOX genes in 16 different abiotic and biotic stress conditions. In general, the MIKC type genes, they were ubiquitously expressed under all abiotic and biotic stresses, whereas nearly 75% of the non-expressed genes, they belong to the 3 M-type families. And interestingly, the genes of all MIKC type subfamilies, they were highly expressed under Fusarium head blight infected spikes and spikelets. In conclusion, our results suggest that MADBOX genes, they are underestimated in the wheat genome, and their exploration might help in understanding the rapid global adaptability of wheat. And cereals, especially wheat and rice, they might lack FLC-like genes, and this could help molecular breeders in identification of alternate targets for fine-tuning of the winter wheat varieties. And the subfamily specific distal telomeric duplications in unbalanced homologs, they facilitate a rapid adaptation of wheat in global environments and points towards a potential mechanism for gene family expansion. And finally, other than their important roles in floral organ identity and grain development, the MADBOX genes, they are active guardians of plants against pathogen insurgency and harsh environmental conditions. In the end, I would like to thank to my colleagues and advisors for their contribution to this work. And I would also like to thank the support we received from Precision Agriculture and Analytics Lab, which is an affiliate of CAS UAF Pakistan, and of course the meeting organizers. If you want to look deep into this work, then it is publicly available at the BioRS preprint servers. Thank you so much. That's all.
Okay, thank you very much for the presentation. Do we have any questions, either from the physical participants or online? Okay, it looks like we don't have any questions. Kasım, thank you very much for your nice presentation. Now we can continue with the next speaker, who is Aska Nuren. Aska will present us about the broad spectrum resistance against multiple PVY strains by CRISPR-Cas13 system in Solanum tuberosum crop.
Hello. Are you Can you see my slide?
Yes, we can see it.
Okay, perfect. Hello, everybody. My name is Azka Noreen. I'm doing research work with Professor Dr. Shahid Mansoor and at NIBG, Pakistan. And I was working with the Professor Dr. Shahid Mansoor on the two projects. The first one is the establishment of the CRISPR-Cas9 system in Solanum tubersum against PBY, and the second one is the development of the resistance against PBY in the Solanum tubersum through the CRISPR-Cas13. And today I'm going to present the development of the resistance against the PBY in the Solanum tubersum through the CRISPR-Cas13A. Okay. The first question is like why we are working on the focusing on the development of the resistance against the potato virus. Why? Because according to the previous data, like in Pakistan, the potato virus Y is the most pathological strain in the specifically in the Punjab region, and those are the potato growing regions. And the interesting thing is about the potato virus Y. It has many recombinant strains, and as well as the many strains and with the recombination as well. And it has like a huge, like the host range families, like not only on the affecting the potato, but also the other Solanaceae family, tomato, tobacco, and pepper as well. And that's why we have selected to basically develop the resistance against the potato virus Y in the Solanum tubersum. And Solanum tubersum is a tetraploid. So it was quite difficult to basically develop the resistance through the classical breeding. And so that's why we selected to use this advanced CRISPR-Cas system to develop the resistance against these pathological strain. For this purpose, like in the in this presentation, we have selected the CRISPR-Cas13 system. As we know, the CRISPR-Cas system is have a lot of the variety and a lot of the variants. The Cas13 is specifically targeting the single stranded RNA, and the PY is a single stranded RNA with the 9.7 kb genome size. So the Cas13 has easily target the single stranded RNA viruses as well. While as compared to the Cas9, Cas12, and the Cas14, they have the ability to target the double stranded DNA, and the Cas14 have ability parallel to the target the single stranded DNA as well. But the unique ability of the Cas13 to target the single stranded RNA, and what we have decided, this is basically the graphical illustration of the project, in which we basically we want to develop the resistance against the multiple strains of the PBY. And the Cas13 basically expressed in the plant, and after that, when it is activated activated by the guide RNA acid, and it become activated and it degrade the viral RNA. So how we have checked the whole strategy? First of all, the PBY genome have the multiple genes, and we have selected and we have targeted the multiple genes by using the multiplex genome editing technique. And for this purpose, we designed the guide RNAs on the conserved region of the PBY. For this purpose, we are able to mutate the multiple sites of the PBY genome, and we got the broad spectrum resistance. For this purpose, we designed the multiple targeting cassette, and to basically to target the HC-Pro gene, colloidal alkyl gene, and PI, and the same VP gene and the P3 gene as well. Those are involved in the specific functions in the interaction with the host protein, in the protecting the viral genome and others. So that's why we have designed a guide RNA on the conserved region to target the multiple sites of the PY genome. And we have developed the constructs by using the golden gateway and the gateway technique and the golden gateway as well. And for this purpose, we basically use the Cas13A with the for the protein expression, we tagged with the flag, and the U6 promoter, Arabidopsis U6 promoter is basically used in this, and the U6 terminator as well with the multiple cassette as well. We confirm the construct by the PCR restriction analysis and the sequencing as well. And after that, we optimize the transgenic stages and from the transformation from the internode to the the development of the plant, we optimized with the different concentration of the acetylserine, zeatin ribosidase, and the indole acetic acid to get the healthy roots and the shoot as well, and to get the higher concentration of the transformation as well. And what we have next do we basically develop the transgenic plant, and we confirm the transgenic plant again through the sequencing as well. And after that, what we have developed, we basically developed the multiple transgenic lines of the Cas13, 1, 2, and the 3 as well. And for this purpose, next we basically initially we targeted with the PVY-O strain, PVY-N strain, and the PVY-NTN strain. We firstly confirmed, as we all know, the PVY don't have an infectious clone. Firstly, we confirmed it through the DAS ELISA, and after that, we infect in the from the source plant. We identify in the source plant, and after that we infect the plants, either the wild type control as well and the resistant line as well. Here you see in the wild type, like the same age of the wild type control and the resistant line as well. They go through the same from the tissue culture stages as well. And here you can see the symptoms of the mosaic patterns on the leaf on the control line as compared to the these are the resistance, while here the wild type control showing the mosaic symptoms. And we basically confirm these are the resistance level through the RT-PCR and the DAS ELISA as well. We perform the DAS ELISA with the 7 days post inoculation, 15 days, and 30 days post inoculation. We collected the samples from the respective plants, and after that we checked it. And the interesting thing what we have done it, we basically do with the mixed. You can set the inoculation with the PVY-O, PVY-N, and PVY-NTN. Here you can see the line Cas 13.2 is more resistant as compared to in all the in the individual assay as well as in the mixed inoculation. And further, we want to basically check the expression of the Cas13 to correlate with our results. We have done the Cas 13.3, 13.2, and 13.3. Here, the Cas13, the expression of the Cas13 is a higher into the transgenic line. So we can correlate this thing like the more resistance in the Cas 13.3 is a more related, positive correlated with the expression of the Cas 13 as well. And next we confirm the PVY titer analysis by the RT-PCR as well to check how much the PVY is accumulated in the leaves after the 45 days of inoculation as well. And what we have done, we perform the same as with the same lines. We perform the RT-PCR on to check the concentration of the virus titer of the PVY-O, PVY-NTN, and the mixed inoculation. And the RT-PCR is also confirmed that the line 13.2 is in the mixed, when we use the mixed inoculation, is also more resistant. So we concluded there the Cas13 is basically the Cas13 is a more resistant. Have ability to degrade the viral RNA, and in the literature they have reported the PVY has the Cas13 has somehow when it become activated, it has a collateral activity, and it has ability to degrade the other RNA as well, viral plant RNA as well. To confirm this, what we have done, we basically target another single stranded RNA, that is the potato leaf roll virus. For this purpose, we basically checked on the plant, and what we have done, we performed the assay, and on the basis of this, we have checked all the lines, either the Cas 13.2, 1, 2, and 3. They have exactly same show the assay after the 45 days of the DPI as compared to the control as well. So we conclude that in our assay in the plants, the Cas 13 has no collateral activity. So it is exactly functional to related to the specific guide RNA. And so what we have concluded, we have concluded that Cas13 have an ability to develop the resistance against the multiple recombinant strains of the POY as we have done it. And secondly, we have confirmed the specific activity of the Cas13 against targeted with the PLRBY. And thirdly, we can conclude and we can assume as we can give the idea and the future prospect as the Cas13.3 is have ability to basically change the RNA basis, and it has the ability to modify the post-translation modification to basically get the more advanced results in the field of the plant. And next, thank you so much for listening patiently, and I'm very much thankful to my supervisor teams, Dr. Shahid Mansoor and Daiba Zainab, who provide me the opportunity to do a research work, and all are my lab fellows as well. Any question?
Okay, thank you very much, Aska. If there are any questions, we can take now.
Yep.
Anything from the online audience? Seems that we don't have any questions. Okay, great. Now we can continue with the next speaker, Manal Ahmed. Manal will talk about development of hybridomas producing map for determining alpha toxin B1 in food. Okay. It seems that I think Manal has left. At the beginning, he was here. Anyways, we will continue with the next person, Shahid Ahmed. If Shahid is available, I think he is also not here. Yeah.
So the name earlier.
Yeah, they both were here, but maybe internet has just gone there. I don't know. Okay, Fahimullah, let me see. But why he is not listening?
Manal, can you please unmute your mic and share your slides? You're the co-host. You should be able to do that.
I think he is not there.
Okay, let's go with the other name.
Yeah. Not here. Okay, we will skip this one. And then we have Fahimullah.
Yeah.
Okay, so our next speaker, Fahimullah Khan. I think he's here, he's available most probably. He will talk about the molecular and field-based screening of advanced bread wheat lines against stripe rust resistance.
Good evening, everybody.
Hi, Fahim, we can hear you. If you can share your screen,
Yes.
You can start your presentation.
Yes. Yes, could you please give me a second? Hi, everyone. I'm Dr. Fahim, an assistant professor of plant breeding at Azadi University, Mansehra, Pakistan. Today, I'll be presenting over small project on molecular and field-based screening of advanced bred wheat lines against stripe rust. As you all know that wheat is one of the most important crop that is grown on wide areas, and Pakistan is among the is on 8th among wheat growing countries in acreage and production of 25.5 million metric ton. Along with the production, we have several issues like we have a lot of areas here in Pakistan. The wheat crop is under drought stress. Other than that, another major issue in wheat is stripe rust, which is also called as yellow rust. So the current study that we did was on screening of our advanced lines that we have developed in our breeding program. There was screen against the stripe rust in 3 locations. The locations were Kohat, Nowshera, and Peshawar. They are diverse location and they have unique races of stripe rust. So we screen our material in Kohat, Nowshera, and Peshawar in field. Coupled with the screening of these lines against the stripe rust, we did molecular screening against. We used some SSR primers for wire 5, 10, and 15. And why we use this primer, why only wire 5, 10, 15? Because in Pakistan so far, these wire 5, 10, and 15 they are like still effective, and any line who are having these genes, they are resistant to stripe rust. So in the current study, we tested 93 bred wheat genotypes, including 3 newly released varieties are the popular varieties, including Kohat 2017, Pakistan 2013, and Morocco is, you all know, is a susceptible check. So we plant them in augmented design and as observational nursery without any replication in all the 3 locations of Khyber Pakhtunkhwa province of Pakistan. So based on the studies, we found that for disease data, we got 5 lines in Kohat, 6 in Nowshera, and 11 in Peshawar. They were having no disease, and 11 lines in Kohat, 1 in Nowshera, and 11 in Peshawar. They were resistant, and 39, 10, and 11 they were like moderately resistant. Similarly, we got 6 lines, 1 line in all Kohat, Nowshera, and Peshawar. They were susceptible to stripe rust. Further that, we did molecular screening using SSR primer, and we got the result for all the lines for both wire 5 and wire 10 genes. Based on the study, we concluded that like both the wire 5 and wire 10 genes are still effective, and they are providing adequate resistance to wheat in Pakistan. And 4 lines, we got some desirable low ACI values without carrying wire 5 and 10 genes, which indicates that there are some unique genes in their genetic makeup, and we are exploring those genes that they are having in the background of those lines, and but they were not wire 5 and wire 10. Similarly, we got that like line KT072, KT007, K2010, and KT062. They produce maximum grain yield. The yield data is not presented here in this presentation. So they were high yielding, and they were also having a desirable low average coefficient of infection values for stripe rust. So these lines could be recommended for exploitation in future wheat breeding programs. We are working on that, and based on this study, we have shortlisted 2 or 3 lines. They are now under testing in the National Uniform Wheat Yield Trials, and most probably we'll be having our new varieties from this material that are presented here that were tested there in that study. With this, thank you so much. I am thankful to HEC Pakistan, the CIMMYT, Hazara University Mansehra for the financial support and other stories in conducting successfully these studies. Thank you so much.
Okay, thank you very much for this nice presentation. Are there any questions? Shahid Mansour has a question.
Thank you very much, Fahim, for presenting a very nice piece of work. My question is that, are there some known negative regulators of resistance against yellow rust. We know that there are multiple resistance genes, but also some negative regulators. So can you comment about this?
Excuse me, sir, your voice is not clear. Could you please type the question in the chat box? I couldn't hear. Can you hear me, sir?
My question is about the negative regulators. Are there some negative regulators of yellow resistance? So, because in genome editing, we are looking for negative regulators as well, so that we can mutate those genes.
We didn't study that, sir. I will look for that about the negative regulators for yellow rust. So we didn't study in this current project, but I will look for in future.
Okay, thank you.
Any other questions from the audience? Great. I think we don't have other questions. Dr. Fahim for the presentation. Now, I think we should have the presentation of Manal Ahmed. Now should be available. So basically the title of the talk is Development of Hybridomas Producing Map for Determining Alpha Toxin B1 in Food. Manal, if you are ready, you can share your screen. Okay. Most early Manal is having some problems sharing the screen. So let's continue with the next speaker. So our next speaker is Siab Alam. Siab will talk about the antimicrobial activity of silver nanoparticles against plant bacterial pathogens phytosynthesized by Mentha longifolia.
My student Jawad is also ready. So.
Hello, assalamu alaikum. Am I audible?
Yes, we can hear you.
Okay, that's good. I am zooming it in, but it's not zooming in. I'm trying to. That's all right now. So hello, assalamu alaikum. My name is Siab Alam, and I have recently completed my BS degree from in biotechnology from the University of Malakand. I am a new researcher, and I am not from the background of molecular biology. I have recently completed my work on my thesis in the nanotechnology field, and I have worked on silver nanoparticles. As the theme of this workshop was different from my field, as I studied on antibiotic resistant strains in the hospital setup. So I actually made this poster presentation on phytosynthesized menthol and nanoparticles against plant bacterial pathogens. So as part of the introduction, pesticide resistance describes the decreased susceptibility of pest population to a pesticide that was previously effective at controlling the pest. Now, the microorganisms generally attain resistance against the conventional pesticides and insecticides due to their excessive use and poor farm management. And this situation is more commonly prevailed in the third world countries like the majority of the Muslim countries, of the OIC countries, which where the situation is getting much more worse. And we are heading towards an era where there will be a complete control of the antibiotic resistant or the pesticide resistant strains. So we will be unable to eradicate the pests that cause infection and that cause problems to our crops, and hence reduce the yield of our major crops. Now, according to the Food and Agriculture Organization, some invasive pests they cost countries at least 70 billion US dollars annually, and are becoming one of the main drivers of the biodiversity loss. According to the Food and Agriculture Organization of the United Nations, the damage from the plant pest leaves millions of people without enough food to eat, and thus it creates the risk of food scarcity and malnutrition and related problems. So the plant bacterial pathogens which cause actions against the major crops in the most of the developing countries. The plant pathogens belongs to various groups like bacteria, protozoa, algae, and some other. So in this very poster presentation, I am much more focused on the bacterial pathogens because this poster presentation is based on the original study performed by the authors of this research paper, study performed in the Qaid-e-Azam University, Islamabad. So, breeding plant bacterial pathogens, the plant pathogens belong to various groups, including bacteria, fungi, protozoa, as already mentioned. So, some of these bacterial plant pathogens are most common and responsible to cause severe actions on the important crop plants. Examples of these are given below is the Pectobacterium keratovorum, which causes onion blight, Xanthomonas oryza, which causes bacterial blight on the rice plants. Also Xanthomonas vesicatoria, which cause bacterial colonies on the tomato and pepper plants, as well as the Ralstonia solanacearum, which colonizes xylem of the plant body. So as we can see here that these are some of the examples we have taken here. These gram-negative bacteria cause infections in these major crops, and thus it causes various reduction in the crop yield. Now, how does resistances develop against these drugs? So there are three major mechanisms by which bacteria create or develop mechanism against these drugs. So one of them is the enzymatic inhibition of the antimicrobial drugs, or we can say that there is some bacteria, they develop the mechanism to produce or to synthesize some enzyme that render our drug or the pesticide unable to treat it or to kill that very bacteria. Now, these hydrolytic and transferase enzyme, another one is the change in the target site. So when we apply a pesticide to a particular plant in order to eradicate a pest, so it is not able to bind to its particular substrate or to its particular site for which it has been designed. The third mechanism, which is more commonly prevalent, is the active efflux of drugs from the bacterial cell walls. So some bacteria, they have these particular housekeeping genes which are specifically designed. These genes encode some pumping proteins. These pumping proteins, they are responsible for the active efflux which involves pumping the antibiotics out of the bacterial cells, which results in the less concentration of the antibiotics inside the cells, and thus renders it unable to kill the bacteria. Now, what we here focused on is the use of silver nanoparticles. So among the other nanoparticles, the silver nanoparticles or AGNPs, they have been widely studied due to their applications in many sectors of life. These AGNPs are mainly used as an antifungal and antibacterial agents. So there are three main mechanisms to synthesize AGNPs, like physical methods, chemical methods, and other biological methods. Here we have used the biological method in which the phytosynthesis of the AGNPs is involved. So Mentha longifolia is a wild plant species. It is from the family Lamiaceae, which is known for its for the essential oils it contains. These essential oils are believed to fabricate AGNPs, and they are believed to help in the synthesis of silver nanoparticles. So the materials and methods used in the study were the biofabrication of AGNPs, optimization of certain parameters like AgNO3, salt concentration, like pH, the incubation time we provide, and then the temperature during the synthesis reaction. Other than that, physical and optical characterization of the AGNPs were employed. At the end, there were the antimicrobial activity of AGNPs in order to assess the antibacterial efficacy of the AGNPs synthesized from the Mentha longifolia. And then the biocompatibility study of the AGNPs was also performed. Now, from the study, we observed that the silver salt concentration, the optimum was 3 millimolar concentration. 60 degrees C was observed to be the ideal or the optimum temperature at which there was much more synthesis of the silver nanoparticle, and these results can be shown here, can be seen here in these peaks. That at pH 9, the SPR band, which is from 450 to 405 nanometer, this is the ideal range for the synthesis of for the confirmation of the synthesis of nanoparticles. Now, the antimicrobial activity of phytosynthesized AGNPs was the phyto-fabricated AGNPs provide an eco-friendly approach to biologically control the plant bacterial pathogens, as contrary to the application of chemical pesticides, which are not eco-friendly, which can cause problems and toxicity in the nearby running water and the environment. So the antimicrobial potential of AGNPs was measured against all these plant bacterial pathogens already mentioned. No growth of bacterial pathogens was observed at higher concentration, and survival percent increased with the lowering the dosage from 500 microgram per milliliter to then subsequently to 250 microgram per liter, then 100 microgram per liter, up to 0.01 microgram per liter, as it can be seen on this graph. Now, the dose responsive antibacterial activity was measured to be between 2 to 12 microgram per ml, which we can say that MIC, the minimum inhibitory concentration observed, was 12 microgram per ml, which means that this concentration was much more much more good to kill the plant bacterial pathogens. At the end, we can say that there was biocompatibility. The biocompatibility of AGNPs was measured to use nanoparticles for any potential human applications. Then hemolytic assay was performed in order to assess the response of the human erythrocytes or RBCs towards the AGNP's application. If we spray that particular pesticide made from the nanoparticles, and if it incorporate into the human food chain, so what could be the possible consequences of the use of these nanoparticles? So hemolytic assay was performed, and we observed that 208 microgram per ml was reported as the lethal dose to lyse 100% population of the erythrocyte, as it can be seen here. But as the dose decreased from 200 microgram per ml, So the lethal dose decreases as we can say that 174 microgram per ml. So the lethal dose was 90, and when 34 microgram per ml was used, so the lethal dose decreased down towards the 50. The colloidal AGNPs were found as non-cytotoxic at a very small dose below 15 microgram per ml. So the dose required for minimum inhibitory concentration was 12 microgram per milliliter, and the cytotoxicity can be observed above 15 microgram per ml. So we can say that it is much more conducive to be used in any future in any possible field study or in the possibly in the field to kill the bacterial pathogens. And it can be very conclusion that these results greatly endorse the use of plant synthesized silver nanoparticles to biologically control the proliferation of plant pathogenic bacterial strains, and the consumption of plants received treatment may not have any harmful effects on the humans because of their compatibility against RBCs. Thank you so much.
Okay, thank you very much for the presentation. From the online audience, we have one question. I would like to take it. We have that question: how do you prepare different doses of this silver nanoparticle solutions? What buffer or solvent do you use?
Actually, there are so many ways to prepare concentrations. Like one way is we compare our concentrations with the way we use preparation, the way we may prepare antibiotic doses, like 250 microgram, like 250 mg, like 500 mg. So in this way, we can we prepare different doses of the antibiotic, and then apply that to the bacteria on the different methods like this diffusion method. So from the minimum inhibitory concentration, we can say that whether the bacteria can be killed or not. And from the biocompatibility assay, we can say that whether it is safe for use in humans or not. So normally, instead of the buffer, we use distilled water, or the distilled water is most commonly used to prepare different concentrations.
Okay, thank you for the answer. Do we have any other questions?
Very interesting work. So have you tested that, this also kill, whether it also kill, friendly microbe, because, probably the mechanism is of general antimicrobial activity, or how we can make sure that, this microbiome, useful microbiome is not killed by these nanoparticles?
Sir, as far the useful microbiome is concerned, so we then refer mainly towards its use in the human use. I am myself currently working in the nanotechnology lab on AGNPs and its use instead of the instead of the traditional antibiotics, the conventional antibiotics. So there we combine the silver nanoparticles in its synthesis phase with the antibiotic. So the lower dose of that antibiotic to which the bacteria is resistant, along with our AGNPs, so they have much more good results than the antibiotic alone. Apart from that, if we are using some broad spectrum antibiotic, it can cause problems because it can kill the friendly microbes in our gut and the bacteria in our gut. So when the dose is decreased by combining it with the AGNPs, so I think it will be more safe to be used than the broad spectrum antibiotics which are currently which are being used currently at a very severe doses in our part of the world.
Good, thank you very much. And have you tried it on fungal pathogens?
No, sir. I am currently applying it on the certain gram-negative strains of the resistant pathogens, bacteria.
Thank you very much.
Okay, thank you very much for the presentation. Now we can go back to Manal Ahmet. He's waiting to share his screen. Manal, do you think you will be able to share your presentation? No. I think no. Where is Jawad?
Probably not in the list, but
Okay. So then we can continue with Jawad. Jawad, can you share your screen? I actually don't know, sorry, your title. It's better if you just start talking. Yeah, he raised his hand to just do the presentation, but he cannot share, I think, the screen. Now, Mana said that, okay, could we give permission to Jawad? Can I do? I don't know.
so the one is ready. So you just need,
I think we cannot do that. Okay, now they did that. Okay, now I think Jawad, you can share your screen.
शेयर ये इसको क्लिक करेगा उस पे.
Okay, now I think Jawad, you can share your screen.
Hi. Hello, everyone. Can you hear me?
Yes, we can hear you.
And my screen is visible, right?
Yes, yes, we can see.
Thank you. Hello, everyone. Myself, Jawad. I'm from Nibsi, Faisalabad, Pakistan. And thank you so much for the opportunity. And the work I will present is more like a perspective, and it's in experiment phase right now. So the work that I'm conducting in my PhD is based on integrative approach, including breeding and CRISPR-mediated genome editing in wheat to enhance the yield. So as we already know that wheat is the staple crop of many countries around the globe, and it contains many essential components like carbohydrates, proteins, and a very good amount of fibers and vitamins that are very crucial for the health as well. And but right now we are facing a dwindling decline with every year. There is a decline in the production of wheat owing to certain climatic changes and other prospects. And due to that, we need some fast-forward technique to cope these problems. And for that, we have tried to combine some certain trade-off traits. The trade-off traits, like maybe you guys know that these are the grain number and the grain size. That is also the weight of the grain. So if we can combine the both trade-off traits, then we can increase the yield to a pretty much extent. So for that, we have to enhance the yield, we have found out some genes that are involved in the grain that can be can down-regulate and can knock out using CRISPR-Cas technology. And these genes are mostly negative regulators of either grain yield, grain number, or grain size. So These genes are the gene that is mostly involved in the grain weight. That was the first target, CRISPR target, and that is TAGW grain weight 2, and that coat a ring type protein that is involved in the ubiquitination process. And the other gene is from CKX family that codes cytokinin oxidase and dehydrogenase. This protein degrades the cytokinin, that is a very essential hormone during grain filling and grain elongation during the process seed maturation. And the other gene that has been recently reported, and it's an ortholog of rice, that is D27. And this gene is basically involved in the number of pillars. And by knocking, down regulating using RNAi, it has been inferred that this gene is involved in the number of pillars, and if we don't regulate or knock down the expression of this gene, then there is an enhanced number of pillars. So by choosing them, choosing these genes, we have designed our multiplexing approach involving CRISPR-Cas9 in wheat, and we have there are for the audience, there are 3 mechanisms that are being used for multiplexing technology, and those involves as you can see in the bottom figure and on your left side, there that involves the expression of all the guide RNAs under different promoters and terminator. And in B, you can use a combination of guide RNA and a tRNA in a chain form. You just have to clone them under just one promoter and one terminator. And the third approach is a repetition of the ribozymes in between the guide RNA, and the ribozymes that are mostly being used are hammerhead and the hepatitis delta virus. They has the capacity to excise them after the transcription. So the strategy we chose is illustrated in B section. We used the chain that will be transcribed in the form of guide RNA and tRNA, then again the second guide RNA and tRNA, and again the guide RNA and tRNA. So we have used this strategy, and we have opted guide RNA from all the homologs from the conserved regions and from various exons, because owing to the repetitive sequence in wheat, we have to be very cautious during designing the guide RNA. Anyway, it's not that complex anymore, owing to the availability of the genome that is freely available. So we have designed these guide RNA on the conserved regions. And as the problem was with the regeneration efficiency of wheat. So recently, as Dr. Haita mentioned, they have optimized that regeneration efficiency technology as well, and they have achieved up to 93% of regeneration in wheat. And so we have already established a vector in our lab that we were using. So I cloned that regenerating cassette from that construct, and we have cloned it in our optimized vector. So these were the material and methods and for the proof of concept, that this is very important, for the proof of concept that the trade-off traits like the number of grain and the size of the grain cannot be achieved in a single generation. we crossed two wheat varieties. One is TD1 that had the grain size, greater grain size, and the other two varieties that we used have high grain numbers. So in F1 we achieved marvelous results, and those results show the average number of grains greater than the parents. So as it was the proof of concept, that we inferred that these traits can be combined together. We can achieve the grain number and grain size in a single line. The results, and we also have enhanced the hybrid vigor was very important, and we have the increase in the number of tillers, the flag leaf area, and all the sentient components were analyzed, and the F1 cross had all the best results, and showing that the hybrid vigor can be achieved in that. So here are the optimized tissue culture protocol that we were using in our lab, but we are now moving towards the tissue culture regeneration tissue culture protocol that is provided by Dr. Haita. And you can, in the bottom left section, you can see the construct that we have developed using the gRNA and tRNA in combination. So the final results, the sequencing is pending, and we are waiting for the results. But from the available data, I have inferred some conclusions and future prospects that can be used in future strategies, and that shows that F1 and the trade-off traits were combined in F1, and by using multiplexing approach, you can target more than one gene simultaneously, and they can I have displayed just the data about the yield, but you can also jump in with the guide RNAs for the resistance and other susceptible genes. And owing to recent advances in tissue culturing and speed breeding technology, the integration of the edited lines can be very much feasible in due time. And recently the advent of a mini CRISPR effector, that is Cas12F1, and merely have a size of approximately 1300 nucleotides. You can use and clone that into viral vectors, and those viral vectors can be used for a non-transgenic approach. And I'm pretty sure it will show excellent results because it has been utilized in mammals and other individuals. So the hindrance in the cloning for Cas molecules has been eliminated due to this recent Cas factor. So I'm positive that we can use this vector as well for from tissue culture free and non-transgenic plant development. Thank you so much, and again, I am thankful to all the dignitaries and the respected audience. And I'm working under the supervision of Dr. Shahid Mansoor and Imran Meen, and I'm happy they are there. And thank you so much, everyone. If you have any question, please go ahead. Can you hear me? Hello.
Thank you very much. Jawad. We can take.
Hello. Hello. Got it.
So,
So I think Priya will be better because we have virtual audience. Dear participants, thank you very much for being here with us. I would request His Excellency, Professor Dr. Iqbal Chaudhary, to please say a few words about the event.
Auzubillahi minash-shaytanir rajeem. Bismillahir-rahmanir-rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. First of all, let me thank all of you for staying on the second day till the late evening. And I understand there are more presentations. So certainly this reflects the interest, this reflects your commitment to the subject. I'd like to begin by thanking the most important people in organizing this event, and these are the organizing team of Dr. Emre Oksay and Hazima Muazzam, There is an outstanding team of people who have been working to make this event happen, and they have done it with excellence and style. I would also like to thank very distinguished speakers. I was here for half a day yesterday, and I could realize that there were some very eminent speakers who contributed in this important event, and this certainly reflects that despite all the problems, there are people who are there to help others and to develop capacity of people. A special thank to my dear brother, younger brother, Professor Dr. Shahid Mansoor. He is an outstanding scientist, a very good manager, and certainly a very high caliber academician. He has not only contributed in Pakistan in the quality of research, but also globally. He's also actively engaged with the ICGB and many international forums. So thank you, Brother Shahid, for your contribution. You certainly have played an extremely important role. And of course, sponsors of the event, like to say, COMSEC, this standing committee, our sister organization for economics and commerce, have been very generous in organizing these events with us, and we hope to continue this partnership with them. Our brother Fateh is not so well. Hopefully, I would be able to see him before I leave, but even if I don't see him, my thanks and gratitude would also be conveyed to him. I had a wonderful meeting with many people, including Rector of the Middle East Technical University, and Dr. Mustafa has been a great supporter of COMSTECH. It just happened that he was the recipient of COMSTECH Prize, and so he knows about COMSTECH, and he constantly follow the activities and initiatives of COMSTECH. So I'm extremely grateful to me to their collaboration and hopefully And what we have discussed that initially 3 countries, Turkey, Kazakhstan, and Pakistan, would take the lead. Of course, we'll invite people from the other places also. The first event of this very innovative idea of OIC Youth Forum, Science Youth Forum. The first of its meeting would be held in May 2022 in Pakistan, Islamabad, inshallah. Theme, our team will decide. Imran will take lead from Pakistan side, and they will decide about the theme of this event. So we would like to have this genesis of a new initiative which will focus on the younger people who would work together with each other, develop and cultivate relationship with each other for 30, 40, 60 years. And inshallah, this will harness the long-term collaboration. These are some of the proposals. And with this, I would like to thank you all for your interest, traveling from different countries. And at the same time, I think what you have done is, despite the fact that there are still problems, Omicron is a new emerging threat also. Hopefully will not commit the same mistake as we have done in case of the first Delta and Beta and others. We would still have an open world. Despite all of these problems, we have traveled, you came all the way and participated in this very successful event. I would also like to thank that you must have been feeling really deprived of wonderful food of Turkey and excellent weather. The Turkey is one country where you can come only for culinary delight. You can just come only for eating. The food is so good. But the fact is, science is as good as anything else. This country has certainly a leadership role in science and technology in the OIC, and we're looking forward to have many of these things coming forward. Move forward. Thank you all, and may Allah subhanahu wa ta'ala bless you with health and happiness. Assalamu alaikum wa rahmatullahi wa barakatuh.
Okay, Manal, you can share your screen, I think. Can we make Manal co-host? Then maybe he will be able to share. Okay, now Manal, can you try to share your screen? Manal, can you hear us? Because we also do not see your microphone. It seems that your microphone is not available. Okay, other than Manal, do we have any other presenters available online? Okay, so I think because we have a technical problem happening in Manal's computer, it seems that we cannot get his presentation online. And it seems that there are no other presenters available. So I would like to end this poster session. And this will be actually the end of the entire lecture part. So tomorrow, we will meet again at 9 o'clock. First, we will have a presentation by Shahid Mansour. After listening to his talk, then we will explain you about the details of the workshop. Basically, we are planning to have a general discussion about the last 2 days. And we are planning to have take the notes of those discussions, and then we will turn it into a final report. And we need to send this report to the COMSEC. This is one thing. And during the discussions, if we do not have any technical problems for the online participation, we can continue with the Zoom. Otherwise, we can skip that. And I think the physical participants would be helping us to generate the report. Okay, I think this is the end. Thank you very much for joining us virtually as well as physically.